Yes, we are live on YouTube. Hello, ladies and gentlemen. Welcome to our International Astrobiology Conference. I am the Vice Director of Molecular Biology and Genetic Turkey social media platform and the leader of the Agenastro team. Today, we will welcome so many precious speakers. Our conference will be held in English. Now, our team member Zeynep will moderate the first session.
Hello, it's Zainab speaking from Agenastro team of Molecular Biology and Genetic Turkey. Welcome to our first International Astrobiology Conference. As you know, we will discuss astrobiology with one more valuable professor than the other. Is everyone ready to start? I can hear your answers as yes. Shall we start with our first speaker, Dr. Tenijia from Japan? He is special appointed assistant professor at Earth Life Science Institute at Tokyo Institute of Technology. Besides, he is an affiliated scientist at Blue Marble Space Institute of Science. He is studying how life is emerged on Earth and can be emerged on other planets. Today, his topic will be from the origins of life to the first membranous cells. Welcome, Dr. Gia. In Japan, it's evening, right? So,
Yes, it's around 5 p.m.
今日のイスタンブルの天気はいいです。日本の天気はどうですか？ご機嫌いかがですか？
Very nice.
Yeah.
So, thank you so much for inviting me today. I'm very happy to join this event, and hopefully, everyone watching will be able to learn a lot from my talk and also everyone else's talk as well.
Yeah, thank you so much also. Would you like to give us some more details about you and your works, Dr. Gia? And then I will appreciate if you can start your talk. Microphone is your turn. Thank you.
Yes, sure. So actually, my name is Tony Jha. I'm a researcher. I'm a specially appointed assistant professor at ELSI, the Earth Life Science Institute at Tokyo Institute of Technology. I'm also an affiliated researcher at Blue Marble Space Institute of Science. And I can give a little bit about my background. At first, I studied chemistry and business economics and management at Caltech. And my research was focusing on proteins and lipids. And then I next did my PhD at Harvard, where I studied RNA sequencing and RNA polymerization. And finally, my interest in understanding all of these biomolecules, proteins, RNA, lipids, my interest in understanding how each of these biomolecules emerged and evolved led me to ELSI in 2017, where I have many projects related to origin of life and astrobiology through chemistry, biochemistry, and biology. And just here's a little brief description of some of my projects. They're very diverse, so please, if you have any questions later, I'm happy to discuss later.
Okay, Dr. Jha, you may start your presentation. Thank you very much.
Great, thank you very much. And so, first of all, I'd like to present roughly how today's roadmap will be. First, I will explain a little bit about what I think astrobiology is. It's a very diverse field, and maybe many people have very different definitions. Then I'll introduce about have a general introduction about primitive protocells, and then I'll discuss the importance of compartments in origin of life through introduction of membraneless compartments and liquid-liquid phase separation. And finally, I'll give 2 case studies in origin of life research through some of my previous and recent research projects. So generally, what is astrobiology exactly? And if we look back into the word, we can see that there's an astronomy, and biology, so life science. So astrobiology includes some aspects of intersecting between astronomy and biology. And in particular, I believe that there are many aspects of astrobiology. For example, the active search for life in space. This is through various space missions, so searching for signs of current life or former life in space. For example, maybe some of you have heard of the JAXA mission Hayabusa 2, which recently came back from an asteroid and took samples back And so we can understand about the possibility of life in space through analyzing samples taken from other extraterrestrial objects. Then, of course, the active search for habitable planets in space, and this is often through searching for exoplanets or probing the habitability of planets and exoplanets through observations on, for example, telescopes or radio telescopes. And then, of course, the active search for the origin of planets, Earth, and life on Earth. And in particular, on Earth, there are a number of different ways that we try to accomplish this. One is to try to search for organisms that live in extreme environments, because the ancient Earth may have been a very extreme environment with different temperature or different salinity. The active research on how planets and geology form, and then some of what I'm interested in and some of what my research focuses on is the search of, for example, how to make living systems in laboratories so we can understand what minimal components of biology are required for life, the search of how primitive chemicals that were present on early Earth assembled into primitive life, and of course, how this primitive life evolved into modern life. And so, as one can see, actually, origins of life, the topic that I'm interested in, is a very small part of the astrobiology field, but also because astrobiology is so wide and complex, extensive collaboration with a number of different scientists, engineers, and even research, and even people from non-scientific backgrounds is essential for accomplishing goals within astrobiology. So let's continue on, and I'd like to introduce a little bit about primitive cells. So for those who are familiar with biology, cells are a unit of life. All life is made up of cells, other than viruses, but that's an uncertain, a gray area. And basically, the cell is a very complex system that accomplishes many specific roles through organelles and other processes. We've learned a lot about modern cells and how they function and their structure in the last 10, 20, 50 years. But one of the questions that still remains unanswered is how exactly the cell emerged and evolved in the first place. One hypothesis is that before the emergence of a modern cell, a primitive cell or a protocell was the form that that was present that eventually evolved into the cells. And this protocell may have contained a compartment boundary, a genetic system, some catalysts, and then presumably this protocell could also undergo evolution, growth, and division. And so one of the goals of origins of life research, there are many goals, but one of them is to understand how the chemistries available on early Earth, we say prebiotic chemistry, how these chemistries, how these chemicals, how these reactions could assemble into a protocell, into a primitive cell, and then how this primitive cell or protocell could eventually evolve into a modern cell. And in my case, I approach this question from two ways. I personally am very interested in the structure and function of this primitive compartment, this protocell. And so one direction is to try to understand how the cell could assemble from prebiotic chemistry. Another one is to take lessons from modern biology and to reverse generate a primitive compartment. And so today I will eventually give two examples of research which attempts to reconstruct this primitive cell or protocell through both of these mechanisms. So let me describe first the first, building up from the top down, essentially going from the modern cell or modern biology back and reconstructing a primitive compartment. So this is a very simplistic model. So if this is too simple for you, I apologize, but I wanted to simplify some of the processes in the cell, which are very complex. And so one of the processes that I would like to highlight here is called the central dogma. And this process occurs in all cells. And essentially what it is the DNA, the genetic information of the cell, is replicated through the help of proteins. Through different proteins, this DNA is transcribed into RNA, and this RNA is translated into proteins. Of course, each step is facilitated by proteins. And generally, this process, the central dogma process, is present in all cells. So although this process is present in all modern cells, what about an early cell? What would this process, or would any part of this process even be plausible in an early cell? Well, it's very likely that the proteins that facilitated some of these processes did not exist. They were too complex and may not have been able to be synthesized or fold into the correct structures. It's also predicted that perhaps DNA was not available in the early cell. And so in an early cell, one hypothesis is that RNA was the first primitive biomolecule that existed in an early cell. And this leads to something called the RNA world hypothesis, which maybe some of you have heard of. And this hypothesis postulates that RNA was the first biomolecule on early Earth because it can store and pass down genetic information because it has a similar structure to DNA. And also RNA can catalyze chemical processes similar to proteins when they fold into specific structures called ribozymes. While we have understood a lot more about the plausibility of an RNA world and also the structure and function of prebiotically relevant RNA, there are still a number of outstanding unanswered questions pertaining to the relevance of RNA in prebiotic chemistry and early evolution. For example, how could monomers be synthesized prebiotically? How could polymerization occur without enzymes? How could denaturation of RNA strands occur after duplex polymerization? And of course, how could we protect this RNA from hydrolysis from the intense and sometimes very harsh early Earth conditions? And so this has also led to some recent research and other theories which posit that instead of a pure RNA world, perhaps there was, for example, a hybrid DNA-RNA early Earth, or even an RNA peptide early Earth, as shown through something called a peptide RNA world theory. And of course, this theory comes from the fact that the ribosome is a dense structure filled with RNA and proteins that may have evolved from simple peptides and RNA interacting together. And so given that it may have been possible for RNA to have coexisted with DNA or peptides or other similar types of structures on early Earth, maybe an early cell could have encompassed some of these molecules, not just RNA. So for example, DNA or peptides or of course RNA. The next question about this early cell is, what exactly would this boundary be composed of? Why is this boundary important? And one thing that is very important for primitive systems is compartmentalization. Compartments can concentrate and segregate reactants. They can promote chemical reactions. They can perform catalysis. Sometimes they can protect molecules from degradation and also prevent diffusion of genetic and other materials. And so compartments on the early Earth are very important for the survivability of a primitive system. In particular, a modern cell, shown here, has a membrane, a bilayer lipid membrane composed of phospholipids, shown here. And although, however, a primitive cell or protocell may not have had the machinery capable of synthesizing these phospholipids that can compose this membrane. And so it's thought that perhaps rather than a phospholipid bilayer membrane, a primitive membrane-bound protocell would have been encompassed by a fatty acid bilayer membrane, shown here. Then the next question that naturally comes is, for example, if we believe that fatty acid membranes eventually evolved into phospholipid membranes, did something actually precede fatty acid membranes? Did some type of boundary exist before the emergence of fatty acid membranes in protocells? And so this then again begets this question of how exactly could this boundary have formed, what this boundary would have been made of. And so that's another question that we will hopefully, well, maybe there's no answer right now, but we will explore later. First of all, I'd like to discuss a little bit about peptides. And so peptides and amino acids are the monomer structures of peptides, and amino acids were likely available on early Earth through, for example, meteorites or atmospheric discharge. And peptides could have also assembled on early Earth through other geological processes, such as, for example, volcanic outgassing or catalysis by mineral surfaces. And so this is a list of perhaps some of the sources of amino acids and peptides on early Earth. Recently, there have been various space missions that have detected peptides. For example, Hayabusa 2, some analysis will happen from the samples that are returned that will focus on detecting biomolecules such as amino acids. And another mission by JAXA called Tampopo has studied the ability for amino acids to conjugate into peptides through cosmic radiation. And so this is an ongoing mission that some of my colleagues in Japan have been participating in. Furthermore, meteorites such as the Murchison meteorite has shown the ability to harbor amino acids. So this might have been one way for amino acids to have been delivered to Earth. And of course, once delivered to Earth, it's possible that mineral surfaces could have catalyzed polymerization of amino acids into peptides and followed by their self-assembly. These peptides are actually very useful. For example, peptides can catalyze vesicle growth. Vesicle is one type of compartment that has been proposed to be a protocell model. And so specific short peptides are able to catalyze vesicle growth. Peptides can also localize specific molecules to these vesicle membranes. In particular, this is research by one of my former colleagues, Neha Kamat, and they showed that an amphiphilic peptide can actually bind to both RNA and to the membrane bilayer surface, thereby localizing RNA to the membrane bilayer. And of course, peptides can actually create compartments. And so one specific type of compartment that can be composed of peptides is a coacervate. So these coacervates are often produced from electrostatic interactions between oppositely charged polymers, as shown here. This type of coacervation is called complex coacervation, and one can imagine, for example, binding of a positively charged peptide with a negatively charged peptide, or a positively charged peptide with a negatively charged nucleic acid like DNA or RNA, forming this structure. And so shown here, these membraneless structures, these membraneless compartments are visible through microscopy, for example. And this type of compartment assembly happens through a process called liquid-liquid phase separation. So phase separation occurs and it results in a soft condensed matter-like phase. It forms droplets that are membrane-free. They don't have membranes, but they're still compartments. They can hold and compartmentalize molecules. These membraneless droplets could facilitate exchange of other molecules between the phases, and in some cases can be formed from simple molecules, which is very interesting for origins of life in astrobiology research. Some of these simple molecules, of course, could be nucleotides or amino acids. And finally, maybe some of you have heard of liquid-liquid phase separation as it's used in drug delivery or chemical reactors, or even in biological phase separation. In fact, modern cells actually contain membraneless organelles, which are formed from liquid-liquid phase separation. And these membraneless organelles, such as chromatin or stress granules, may actually have some link with primitive phase separation as well. So there's two types of phase separation. One is non-associative and one is associative. And so the non-associative type actually occurs when, for example, mixing two types of polymers, and these polymers, for whatever reason, are not chemically compatible. Maybe their chemical affinities are different, maybe their polarity is different. And so this results in the segregation of these two polymers from each other, forming a specific type of droplet. And shown here, this is two types of polymers, polyethylene glycol, PEG, and dextran. And when those are mixed in specific quantities, what forms is a dextran droplet within a PEG phase. And this is one example of a non-associative liquid phase separated system, also called an aqueous two-phase system. The other type is the coacervate type, or complex coacervate, which I had briefly introduced before. And these form through the binding of oppositely charged polymers or polyions, and this results in a dense coacervate phase composed of those components, and a dilute phase around, which is composed of very dilute water-based solution, shown here. And these coacervates actually perform a number of very relevant prebiotic functions. For example, coacervates can segregate and concentrate a number of primitive molecules or biomolecules, such as nucleotides, nucleic acids, or divalent cations, which may have been necessary for the synthesis or function of various nucleic acids. Coacervates can also undergo growth and division. For example, agitation or physical forces can result in budding or division of these coacervates. They can coalesce to grow, and also these coacervates can divide through subjection of external forces like electric fields. And finally, coacervates have the ability to increase in their structural complexity. So previously we've shown only single-phase coacervates, but it is possible to construct multi-phase coacervates, shown here. These are two different types of multi-phase coacervates, and these compartments are composed of components that each have their own chemical affinities or their own chemistries, resulting in a very complex compartment that has different characters as shown here. Another way to increase the structural complexity of a coacervate is through membrane assembly. And so it's been shown that coacervates can actually help to facilitate the assembly of a fatty acid or phospholipid membrane around itself. And this is actually interesting because it foreshadows the possibility of the evolution from coacervate or a membrane-less protocell to a membrane-bound protocell. So all of these studies have shown specific functions of coacervates, and the two recent ones that I showed the ability for coacervates to increase in structural complexity. And so one of the studies that I was involved in, one of the initial goals was actually to try to find another way to increase the structural complexity of a coacervate. And so this project was in close collaboration with a faculty member in France, Tommaso Fraccia at ESPCI Paris. And so his expertise is actually in DNA liquid crystals. And one of the goals of this study was to combine DNA liquid crystals with coacervates. So if you're not familiar so much with DNA liquid crystals, maybe you've heard of liquid crystals, for example, in displays or screens. They can actually also be composed of DNA, and this occurs when DNA, in this case, for example, DNA oligomers form duplexes. These duplexes will aggregate through end-to-end adhesion, forming these stiff rods that will stack upon each other. And once these rods start to form into bundles, the liquid crystal phases actually will emerge, and one can visualize liquid crystal phases through a technique called polarization microscopy. And shown here, it's some liquid crystal structures from DNA. In particular, we were interested in incorporating DNA liquid crystals into coacervates for two specific reasons. One is DNA by itself could form coacervates. DNA that is bound to a cationic peptide like polylysine has been shown to form coacervates. And also, DNA liquid crystals, or liquid crystals in general, have been shown to contribute specific functions such as electrochemical selectivity, regioselectivity of chemical reactions, or even the promotion of or catalysis of certain reactions such as polymer ligation or polymerization. And so we sought to combine a known DNA that can form liquid crystals and combine that with a polylysine, that's a cationic peptide, a positively charged peptide, and to see whether we could co-assemble a DNA liquid crystal coacervate structure. And so through a number of trials and errors, we were able to find specific conditions that resulted in the co-localization of the liquid crystal phase with the coacervate phase. So seen here, the top is a bright field microscope image, the bottom is a polarization microscope image, and you can see that the DNA liquid crystal structure is co-localized with the membraneless droplet structure. And we believe, of course, the DNA liquid crystal is composed of these short DNA duplexes that are stacked upon each other that perhaps combined with or interacting with cationic peptides can also form these membraneless droplets. So another thing that we determined was that these liquid crystal coacervates were could only be assembled in very narrow salinity conditions. And so here is a titration of sodium chloride, and we could see that at very low sodium chloride concentrations, precipitation occurred. At higher sodium chloride concentrations, actually what occurred is first the disassembly of the liquid crystal state, followed by disassembly of the coacervate state. And we believe that this is because introduction of sodium chloride actually results in shielding, and this will result in the modulation of the electrostatic interaction between the peptides and the DNA. For example, at lower salinities, the binding between peptide and DNA is much stronger, forming these precipitate structures, whereas at very high salinities, the binding is very weak, and between the DNA and the peptide is very weak, resulting in eventual disassembly of the structures themselves. Another aspect we were next interested in studying is how important this end-to-end DNA duplex stacking interaction was in forming these structures. And so we actually designed a DNA which was the exact same sequence, a 12-mer, but with a 2-base overhang, TT, so that although the DNA could interact with the peptide, it could not interact with other DNA in the same way and could not stack. And in this study, we observed that in all of the conditions we tested, although we could see membraneless droplets or coacervates forming, we in no cases saw liquid crystal co-assembly. And we attribute this to, of course, the inhibition of the end-to-end stacking interaction that is required for liquid crystal assembly. We actually next calculated the amount of DNA that was up concentrated in the liquid crystal coacervates, and in almost all cases, the amount of DNA within the droplets was 2 to 3 orders of magnitude higher than the DNA outside of the droplets, suggesting that this is actually one mechanism to increase the concentration of perhaps very dilute primitive chemicals. So the next phase of this study, we wanted to subject these liquid crystal coacervates to a number of different environmental conditions that would have been relevant on early Earth. And so the first one we subjected them to is dehydration. That would have been plausible through, for example, evaporation, changes in temperature, through different heating or different seasons or day-night cycles. And so here we started with a system that was in the precipitate state, DNA peptide precipitate, and we let the system dry down. We allowed it to undergo dehydration. And what this video and these images show that is that through this dehydration process, the system actually transitions from a liquid crystal, from a precipitate state to a liquid crystal coacervate state, eventually to a coacervate state with no liquid crystals, and finally to a disassembled state. And this is actually very similar in structure to what we saw in the sodium chloride titration. And so this led us to believe that perhaps the dehydration process is modulating the salinity of the solution, and this salinity modulation actually transitions the system through each of these different phases, similar to the salinity titration. The next plausible prebiotic environmental change that we investigated was heating and cooling. And so we first started with, again, a precipitate structure at room temperature around 20 degrees, and we heated this structure first to 50 and then to 60 degrees. And at 60 degrees, we observed that the structure transitioned into a liquid crystal coacervate droplet state. Further heating resulted in disassembly of the liquid crystal state, but the membraneless droplets, these coacervates, were still present. Then we proceeded to drop the temperature, and upon decreasing the temperature to 40 degrees, we noticed that there was the appearance and coexistence of a number of different liquid crystal coacervate structures with the membraneless coacervate structures. Shown here, there's actually 2 different types of liquid crystal phases, which I won't get into the details, but basically a number of different liquid crystal coacervates appeared in coexistence with the non-liquid crystal coacervates. And upon further decrease in temperature, we observed that most of these coacervates actually transition into a liquid crystal coacervate state, as shown here, especially after decreasing the temperature to 10 degrees. And what is really interesting here is that, in fact, the system was different at roughly the same temperature after heating and cooling. So we believe that heating and cooling, or this is perhaps otherwise can be known as an annealing process, actually allows the system to access its most stable conformation, perhaps the liquid crystal coacervate state. And in fact, we believe that this heating and cooling process, or this salinity process, actually also affects the internal structure of these liquid crystal coacervates. So from the left here, at low heats or low salinities, the DNA in blue interact with the peptides in red, and the blue rods, the DNA, actually will bundle together into these hexagonal shapes, forming liquid crystals, co-localized with the coacervates. Increasing heat and salinity starts to disassemble these bundles, and so the structure of the liquid crystals change. Further heat and salinity will result in disassembly of these long DNA rods, although the peptide DNA interaction is still present, and so this results in the assembly of non-liquid crystal droplets. And finally, at very high heats and salinities, the DNA and the peptide actually disassemble completely, and they no longer interact, resulting in disassembly of all structures. So we actually observed that modulating the salinity and the temperature could result in these structural changes, and we also observed that these structural changes were actually reversible. So even if you increase or decrease the salinity, as long as it goes back to its original state, generally after the first annealing cycle, the structure can be reassembled. So some of these studies are published already. So if you have any interest, I encourage you to take a look at this study, which was published last year, and also this study, which was published last year. There's a lot more experimental details for your interest if you want to know more about those. So now let's go back to this initial theme of building this compartment from both directions, and let's focus on how we can build this compartment from the bottom up. So I've mentioned before that there are a number of ways by which biomolecules could have formed on early Earth through primitive geologies, for example, meteorites or atmospheric discharge. Well, actually, at the same time, although these biomolecules formed, there are actually also a number of other molecules that are formed at the same time that may not be considered biomolecules. For example, in the Murchison meteorite, although we can detect amino acids, some of these biomolecules, the majority of the compounds that exist are actually not biomolecules. Similarly, the Miller-Urey reaction or atmospheric discharge, although it produces certain biomolecules like amino acids, it also produces a number of other molecules that may not be considered biomolecules. And we actually believe that rather than asking the question, how did biomolecules form on early Earth and how did biomolecules initiate the first life on Earth, could non-biomolecules actually catalyze or help to contribute to life's emergence on early Earth? One of the interesting non-biomolecules that we like to study are alpha hydroxy acids. And I know that hydroxy acids are present in biology, but they are not present in the central dogma. And so here we refer to them as non-biomolecules, as they are not, generally, they're not lipids, they're not peptides or amino acids, they're not nucleic acids or nucleotides. And so hydroxy acids are similar in structure to amino acids, except an amino group has been replaced with a hydroxyl group. These hydroxy acids can also form polyesters very easily through dehydration. And so through simple dehydration, these alpha hydroxy acids can actually form fairly long polyester polymers through just simply drying a solution. It's actually been postulated that hydroxy acids were precursors to amino acids in that forming a depsipeptide intermediate, which is combining hydroxy acids with amino acids and conjugating them. Doing this process of drying and wetting, this dehydration-rehydration process a number of times actually results in formation of peptides. Furthermore, some research from about 15 years ago has shown that rather than amino acids, tRNA can actually be charged with hydroxy acids, alpha hydroxy acids. And these alpha hydroxy acid charged tRNA can actually undergo translation, forming polyesters instead of peptides, suggesting the compatibility between hydroxy acids and modern biology. And so given this research, we postulated that perhaps we could use polyesters and hydroxy acids as a way to form a combinatorial polymer library, for example, starting with a monomer set of hydroxy acids undergoing drying or wetting and drying cycles, resulting in a polymer library of polyesters. And one question is, could these polyesters assemble into something useful for life's emergence? And so what we did was we took a number of different hydroxy acids shown here. We subjected each of them as pure solutions to dehydration, and we analyzed the products. And shown here are the synthesis products of these hydroxy acids after drying. So I'll zoom into one. So this is a mass spectrum. And what this shows is that after drying of lactic acid solution, what forms is a polydispersed mixture of polylactate polyester of different lengths from 1, 2, 3, 4 monomers up to around 40. And so this polydispersed polymer product is actually observed in all of the different polyester systems that we observed here. What also we noticed was that after dehydration of each system, what resulted was a gel-like structure, and this was observed in almost all of the structures. So after synthesis, the polyester, these polydispersed polyesters actually form a gel-like structure. And we rehydrated these gel-like structures, and it turns out they form a cloudy solution. Usually a cloudy solution could be indicative of suspended particles in the solution. And so we took this solution and we subjected it to microscopy, and we found that in many of these cases, what formed were membraneless droplets. These droplets are formed from these polyesters in solution, and a number of different polyester chemistries could still form these droplets. In fact, we combined, of these 5 hydroxy acids, we combined 2, 3, 4, and 5 of them in all combinations, subjected those solutions to drying, and then we analyzed the products by microscopy, and we observed that in almost all of the cases, these droplets formed as well. And so we believe that these droplets are non-associative. For example, the polyesters are in a polyester-rich phase surrounded by a dilute aqueous solution. And so what you should know is polyester gels assemble into microdroplets in aqueous media. These droplets are actually very interesting, as they will coalesce on a fairly long time scale over hours or days, but on a shorter time scale, these droplets don't coalesce very rapidly. Sorry for the poor video quality here. However, upon adding sodium chloride, we noticed that these droplets actually could coalesce more readily, and actually the droplets would also attach to the glass surface more readily. And shown here, you can see these droplets coalescing much more quickly than before through addition of sodium chloride. And this is also true when adding a buffered solution to change the pH to 8. And so what we believe this is happening is perhaps the addition of these salts could be either changing the droplet surface tension or the droplet surface charge, eventually promoting their coalescence. And it's actually interesting to think about coalescence and how to initiate coalescence through changes in salt or changes in pH. And so perhaps one could envision a system where these droplets could actually recombine upon changes in pH or changes in salt, followed by agitation to result in their division from larger droplets. Furthermore, we subjected these droplets to heating to 90 degrees, and heating to 90 degrees over 5 minutes did not significantly change their structure, although we have done some follow-up studies that have shown that heating of these droplets at 70 to 90 degrees over a few days or a week actually results in their disassembly. And we believe that this may be because the increased temperature, along with the amount of water in the solution, results in the hydrolysis of these polyesters over time, resulting in the disassembly. One of the other very interesting functions that we observed is the ability for the polyester droplets to segregate dyes. Here we subjected 2 fluorescent dyes to these polyester droplets, and these fluorescent dyes, thiophanate T and cyber gold, are actually fairly hydrophobic non-polar molecules, and we believe that the segregation of these dyes into the polyester interior suggests that the interior of the polyester is fairly apolar as well. And so, of course, these dyes differentially segregate and compartmentalize into droplets. And so perhaps this differential segregation could result in a complex or mixed polyester system where certain droplets have certain ability for certain chemicals, whereas other droplets have certain affinity for other chemicals. So here we've described a little bit about how to produce this system from the bottom up. We can assemble these droplets, these polyester droplets, from prebiotic chemistry. And so one of the questions that I wanted to ask was then, could we incorporate some aspects of modern biology into these experiments to see whether these polyester droplets are compatible with modern biologies? And so again, as a review, of course, we see, in modern biology, nucleic acids, proteins, peptides, phospholipids as well. And so we want to subject each of these biomolecule types to the polyester droplets to see for their compatibility. And so at first we were able to, with the help of some colleagues at ELSI, we were able to subject superfold GFP, a protein, into these polyester droplets. And what's interesting to note is that the protein continues to be fluorescent within the droplets, suggesting that in the droplets, the protein still retains a functional structure that allows it to fluoresce. We also observed that fluorescent phospholipids could assemble around these droplets. And so here we subjected rhodamine PE fluorescent phospholipid around these droplets. And although we can observe some type of layer, it's still unclear whether this lipid layer is a single layer, a bilayer, multiple layers, or even lipid aggregates that have apparently assembled around the droplet. And so further investigations into this structure are ongoing. Finally, we subjected RNA to these droplets. And so if you remember a long time ago, maybe 30, 35 minutes ago, we were discussing an RNA world and the origin of life. And so we subjected a functional RNA to these droplets. This RNA is a self-cleaving ribozyme called the hammerhead ribozyme. And we observed, first of all, that the fluorescent RNA could segregate to the droplets. We also observed that in the presence of the droplets, the RNA could still accomplish its self-cleaving function, shown on the right. And so this suggests that these droplets are compatible with lipids and proteins and RNA. And perhaps these droplets still even promote the function of some of these biomolecules within them, suggesting their plausible relevance to being primitive cells or protocells that could harbor functional biomolecules. So one of the questions then is, we saw that only one type of polyester could segregate RNA, polyphenolactic acid. All of the other types of polyesters could not segregate the RNA for various reasons, such as chemical affinity. And so one of the next questions we wanted to ask was, could other polyester droplets compartmentalize RNA as well? And so one of the mechanisms that we chose to do this was to change the chemistry of the droplets. Previously, each of these polyester droplets were composed of purely neutral polyesters. And so we wanted to introduce a cation, a basic hydroxy acid into the polyester synthesis reactions, and then see whether adding this basic residue, this basic hydroxy acid, could result in the formation of a cationic polyester that could form a cationic polyester droplet. And so here we used 4-amino-2 hydroxybutyric acid. From now, it will be shown as 4A2H. And so the first thing we did was we subjected 4A2H in a 1-to-1 ratio and mixed it with each one of these previously used hydroxy acids. So in each reaction, the result was a 50-50 mixture of 4A2H and one of these hydroxy acids. And we subjected each of those mixtures to drying to form polyesters. And so here is the mass spectrometry results of those reactions. And I'll spare you the details, but basically what we observed was that it was possible for 4A2H, the cationic or the basic hydroxy acid monomer, to incorporate into polyesters synthesized through drying when mixed with each one of these neutral hydroxy acids. And some of these polyesters that were formed were up to 12 or 15 residues long. And we observed this for each of the neutral hydroxy acids. So after the synthesis, after we formed these presumably somewhat cationic polyesters, we actually subjected these polyester products to rehydration, and we tried to observe them through microscopy. And we observed that, first of all, most of these structures form gels, like the previous demonstration. Some of these gels, when diluted in water, were cloudy, some weren't. And so this suggested that some of them could still form these droplets. And shown here, we observed that 2 of these mixed cationic polyester systems could form droplets. The 4A2H phenolactic acid and the 4A2H leucic acid systems could still form polyester droplets, whereas the other mixed systems could not form droplets. Some of them formed oils, or some of them formed nothing at all. And we were interested, first of all, to see what could govern the formation of polyester droplets in this case. And one of the first hypotheses we made was perhaps the ratio of the basic residue with the neutral residue followed by drying could actually control whether these polyester droplets could assemble or not. And as we suspected, the ratio of mixing actually is a very important parameter shown here. Going from left to right is actually increasing in the cationic nature of these droplets, or the amount of basic residue incorporated into the droplets. And we see in both cases that at very high 4A2H concentrations, the droplets actually disassemble, whereas at lower or no 4A2H concentrations, that the droplets can assemble. And so we actually performed this study with all of the other systems in a 1-to-1 ratio, and we observed that in all of the cases that lower 4A2H, lower cationic character, actually resulted in more formation of the droplets. Higher cationic character resulted in less formation of the droplets. One of the cases formed no droplets at all, and we believe that at high cationic characters, we believe that the resulting polyesters actually are very susceptible to being dissolved. They're actually soluble in these aqueous solutions. So this suggests that higher amounts of cationic polyesters will result in solvation of droplets, whereas much lower cationic natures, more neutral or more hydrophobic natures of these polyesters will result in more formation or assembly of these droplets. So of course, now we've demonstrated that these droplets can be assembled with some cationic character. The next thing we wanted to test was whether adding this cationic character could actually result in the acquisition of the ability for these droplets to segregate RNA. And so the first demonstration here is with 4A2H and phenolactic acid, which, if you remember, phenolactic acid by itself could already segregate RNA. And what we see here is that RNA, shown in red here, fluorescent RNA, could segregate very strongly to the mixed 4A2H phenolactic acid cationic polyester droplets. And in fact, when we subjected some of the other neutral systems to 4A2H, increasing in cationic character, presumably, we observed the acquisition of the ability for these droplet systems to segregate RNA, suggesting that perhaps the increase of the cationic nature of polyester droplets even a little bit. As shown here, we've only increased the ratio to 20% for A2H. That even increasing a little bit the cationic nature of these polyesters could result in the assembly of droplets with cationic nature that could interact with and segregate RNA. Additionally, one of the other additional observations we made was that actually increasing the cationic character actually could result in the acquisition of autofluorescence of some of these droplets. And we believe that this might be due to the formation of these fairly high molecular weight melanoid type structures that also have the ability to autofluoresce. And of course, we're not entirely sure about the mechanism, and further studies are on the way. And so here I've explained a little bit about one of the case studies involving using polyester microdroplets as primitive compartment model systems. So if you're interested in knowing more about the details, the experimental details, the logic, the philosophy into some of these studies, I encourage you to read some of these papers, which were published. Two were published 2 years ago, and one was published last month. So please feel free to access these online whenever you get a chance, if you have interest in learning more about the details of these polyester studies. So I'd like to take this opportunity to acknowledge my funding sources. I'm very grateful that this research can be funded through a number of different means. For example, Tokyo Tech, my home institute, Japan Society for Promotion of Science, another Tokyo Tech grant, the Japan Astrobiology Center, and in particular, the liquid crystal coacervate work was funded in part by a grant from the French Embassy in Japan, allowing collaborative research travel for a visit to France. And so I'd also like to acknowledge some of my collaborators for these studies. So Irina Mamajanov, Jim Cleaves, and Kuhan Chandru have been some of my very close collaborators working on the polyester droplet work, whereas Tommaso Fraccia is a very close collaborator, and we've been working on the DNA liquid crystal coacervate work. I'd also like to take this time to thank my other colleagues and collaborators from ELSI and also from Tokyo Tech, as well as my collaborators and colleagues from institutes around the world. I think research is really getting more and more to be a very international activity, and having resources and colleagues from various international places has become very important for progression of research. So I'm very grateful for the ability and the opportunity to work with all of my colleagues from around the world. I'd like to take the last couple minutes to briefly overview ELSI, the Earth Life Science Institute, which is my research institution. So ELSI is a research institute at the Tokyo Institute of Technology. It's funded by a Japanese government program called WPI, World Premier Research Center Initiative. And each WPI institute is focused on a specific type of research. For example, some of them are focused on cellular material, some of them are focused on sleep medicine. In particular, ELSI is focused on understanding the origin and evolution of planets, Earth, and life on Earth. And to that end, we actually have a number of different fields represented by the researchers. If you remember, astrobiology is a very interdisciplinary field. And so at ELSI, we have planetary scientists working next to geologists, working next to chemists, working next to biologists, and even working next to computational researchers or theoretical researchers studying complex systems or even artificial life or artificial intelligence. We also have a number of events normally. Of course, this year, most events, and last year, most events have been digital, but hopefully in the future, you have an opportunity to visit ELSI if you're interested. We have an annual symposium. We sometimes host schools, short-term training schools, winter schools, summer schools. And most recently, we actually have started a new graduate course. So sorry for the language in the poster on the left. They haven't made an English poster yet. but essentially there's going to be a new Earth Life Science graduate course at ELSI, at Tokyo Institute of Technology. It's tailored towards 5-year people who are interested in a 5-year integrated master's PhD course. A number of full scholarships are available for international students, and you would have the opportunity to study one of a number of different topics within Earth and Life Science, either through the Life Science and Technology Department or through the Earth and Planetary Science Department. The application deadline will be sometime in October. Of course, the exact date is not yet determined, and more info for international students will be available this summer. And the program would start in April of 2022. So if you have some interest in pursuing graduate studies in the future at ELSI, please feel free to send an email to this address here, or please feel free to email me anytime, and I'm happy to help connect you to the appropriate staff or faculty to help facilitate your educational interests. So again, I'd like to take this time to thank you all for your attention. I'd like to thank the organizers for, again, inviting me to participate in this event. And here I've left my email addresses and my Twitter handle. So I'm happy to take any questions now, but in the case that you don't have an opportunity to directly ask a question now, please feel free to send me an email anytime, and we can discuss later if you'd like. So again, thank you very much for your time and attention.
One of the most questions I have heard from people around me was whether there is a life that can be in a different model or combination that different from we know. So your presentation was answered that. Thank you very much. Thank you for this awesome, marvelous speech and for sharing this valuable information with us. We have many questions from our audience. Now, by your leave, I will guide those questions to Dr. Gia.
Okay, thank you.
Okay. First question from Matthew Salur. Why did the cell membrane evolve?
So that's a very good question. The question is, why did the cell membrane evolve? And the modern cell has a number of, the modern cell membrane performs a number of functions, and one of the most important functions is the ability to generate chemical or electrical gradients. So if you remember back to your molecular biology class, that actually, for example, ATP is generated through a chemical gradient across a membrane through the help of a protein channel. And so it's possible that a membrane was required to generate this type of chemical or electrical gradient for further function, whereas right now it's still not clear whether or how well a membraneless droplet could generate a similar type of energetic gradient. So to answer your question simplistically, perhaps the generation of electrical or chemical gradients facilitated by membranes was necessary for the further evolution of more advanced life.
Okay, thank you for your answer. Now I'm reading the second question. What do you think about XNA? How could the recently discovered XNA molecule have affected the evolutionary process of DNA as we know it so far from NORMAD?
Okay, thank you very much for that question. The question is about XNA. And so I think XNA and other types of nucleic acid like TNA or PNA are also all very interesting. It's very clear that modern biology contains DNA. And so maybe a lot of research is focused on how DNA or RNA develop, but it's also very interesting and very appropriate to study how other types of nucleic acids could function and evolve as well. One of the recent hypotheses is actually perhaps before RNA there was another type of nucleic acid structure called pre-RNA, and so the structure and function of pre-RNA is not entirely clear, not entirely determined, and so understanding about XNA or other types of TNA or PNA, understanding the structure and functions of these types of nucleic acids could give insights into perhaps how RNA emerged and how DNA emerged, even if XNA or PNA or TNA did not or LNA did not directly evolve into RNA or DNA themselves.
Okay, our third question is from Sri Kavya. If I am misspelling, I'm so sorry. I have heard that lipids have been existing long before RNA or any proteins. What possible lipid-lipid interactions could have existed? Are there any studies on this?
Okay, thank you very much for your question. Your question, I believe the question is, so lipids likely existed before RNA, before proteins. So what types of interactions could have existed between lipids and other biomolecules, or generally between biomolecules? And yes, I think it's true that, well, lipids and other types of amphiphils that may not be fatty acids, so other types of amphiphils have been observed in, for example, geological conditions like from meteorites. And so they certainly existed. And RNA and proteins in particular may have existed very late into the development of life because of the requirement of polymerization, and in the case of protein, polymerization, specific polymerization and specific folding. And so it's very likely that these types of structures may have existed at different times. And some of the, however, it's also very clear that modern biology uses a lipid membrane. And so because RNA and lipids and proteins all exist in modern biology and modern cells, it is very likely also that they existed at the same time, perhaps not very early on, but later on in the development of primitive life. And so it is very essential to study these interactions. And so, for example, some research has shown that adding peptides to lipid membranes can increase the fluidity or change the fluidity of these lipid membranes. Some studies have shown that lipid surfaces can actually catalyze nucleic acid or RNA polymerization. And sometimes some research has also shown that short peptides can inject into lipid membranes and result in their growth. And so there are a number of different possible interactions. And I think that now is a really great time to explore and study more of these interactions, not just in isolation, but also in a more complex system. And because we know that the prebiotic Earth was a very complex system, it contained a number of different chemicals mixed together at very different concentrations. And so perhaps understanding about how to analyze and studying these very complex mixtures of primitive biomolecules could also help to give insights into how primitive life evolved.
Okay, thank you. Now another question coming from Anil Turan Karasakal. On the planets we think could be life, it's the cellular level, right? Or we are thinking more complicated lives might be?
So the question is, on other planets, what types of life would we expect? And so the question is, do we expect cellular life or do we expect more complex life? And so, of course, the answer to this is a little bit unclear. However, I do believe that right now many of the space missions are focused not exactly on just detecting, for example, extraterrestrial life, not detecting cellular-based life, but they're interested in developing technologies to detect molecular biomarkers of life. For example, maybe cellular life existed and we can have some type of molecular biomarker to indicate that the cellular life existed. Or other types of missions are really interested in understanding environments, the chemical environments or the geological environments of other planets. And so by detecting the geological environments of other planets, we don't directly detect life or don't directly detect modern or extinct life there. Instead, we detect we can understand more about the plausibility that life could emerge there or could have emerged there in the past. And so one of the things that I think we should all keep in mind when we're thinking about life detection missions and astrobiology-based missions is that we're not only detecting modern or extinct life, we're also detecting environments that could be plausible for future life to also emerge there.
Okay, thank you. Another question from Pelin Küpeli. It's a silly question, but I'm curious about your thoughts on the endosymbiotic theory. It's a crazy theory. Is it possible to experience a similar situation again?
Can you repeat that question?
Okay. She's asking about endosymbiotic theory. Is it possible to experience a similar situation again?
Yeah, so I'm not entirely familiar with this theory. I think that what this theory, so I'm not very familiar, but I believe that this theory basically states that, for example, certain organelles that are incorporated into our modern cells used to be separate organisms in the past. I believe this is the theory. And so in prebiotic Earth, I believe that some type of endosymbiosis would have been possible. For example, the engulfment of a membraneless compartment from a membrane-bound compartment. Something like that could be possible. Of course, much later on, when we have LUCA and, or the multiple LUCA theories, that's also possible. However, one thing we do have to keep in mind is that I believe the endosymbiosis theory assumes that the organisms are all distinct, and before the origin of life, during the protocell phase, it's unclear what could be considered an organism or what can be considered a separate protocell. And so for that reason, the definitions of endosymbiosis on prebiotic Earth would not match directly, but a similar process that resulted in combination of prebiotically relevant or primitive structures or assemblies combining could have been possible. Just maybe that word is not the exact correct word for that process.
Thank you. Another question from Selen Özen. Thank you for the excellent presentation. Any comments on the studies about the role of phosphate present in phospholipids as well as DNA, RNA, ATP, etc., or ribose groups?
Okay, the question is about, thank you. Yeah, the question is about phosphates, and phosphates are, of course, an interesting structure. They actually facilitate a lot of these important biological interactions or structures that we see today, as was listed by the questioner, the viewer. And so we're actually interested in studying other types of potential primitive phosphates, like polyphosphates. For example, could polyphosphates be used as primitive energy sources? Could they form specific types of structures as well? And so some of these studies are ongoing. So wait a while. Hopefully there will be something interesting coming up in the future.
Okay, thank you. And another question, a little bit more personal, but I will read it. From Eda Düzgün, what was the most challenging situation for you while working?
Yeah, so research is it's challenging. It's not easy, but it's also very rewarding. For example, we're able to research topics. Our jobs are to research topics that we find very interesting, or to research into developing tools or technologies that are really advanced, that can help to advance or help to support society. And of course, one of the very important processes of doing research is the funding process. And so during the funding process, often the funders tell, the funders give some funding to do a specific type of research. And sometimes research is constrained by funding. And so, of course, that's somewhat unavoidable because we are at we do rely on funding from government or private sources. And so being able to balance truly being able to experience or pursue research based on interest versus research that was based on a funder's request or a funding mandate is one of the more challenging aspects of research. And of course, recently, of course, last year, we've all been in this COVID pandemic. And so as an experimental researcher, I and other experimental research colleagues as well, we've had a lot of difficulty going into the labs to do experiments. So that's been challenging. And finally, the ability to connect in online conferences has been a very good addition to fill in the gap of the inability to travel personally to the conferences, to physical conferences. And so that has helped to, for example, help to maintain scientific discussion or discourse with other researchers around the world, despite other aspects of our lives or other aspects of our experiments or work being disrupted. So hopefully things get better sometime soon, this year or next year, and we can, go back to being able to do our experiments in the lab, to being able to meet people in person. So I hope that someday I can meet you, all of the viewers, in person somehow as well.
Okay, thank you for your answers, and also thank you for those questions. If I couldn't read any of them, I'm so sorry. Now we have run out of time with Dr. Gia, therefore we need to say Dr. Gia goodbye. I'm very glad to welcome you at our conference, Dr. Gia. Thank you very much for joining us today. I hope to see you later also. Goodbye.
Thank you very much.
And now we will continue with Dr. Armanda-Azua-Bustos after a short break. See you later. Bye-bye.
Hi everyone, welcome to the second session of Molecular Biology and Genetics Turkey, Agenastro Astrobiology Conference. I'm Sude Akalın from Agenastro Astrobiology team. I will be hosting this session, but first, let me introduce you our next speaker. Now, we will be welcoming astrobiologist Dr. Armanda-Azua-Bustos. He is a researcher in Spanish National Research Council and the CEO of Atacama Biotech, which is a well-known Mars analog model at Atacama Desert. To shortly define his work, we can say he is working on life in extreme circumstances. Today, he will be with us on the topic of habitability of Mars and detecting life. Hi, Armando. Welcome. How are you?
Hello. Good morning, everyone, and thank you for your invitation. So,
Thank you so much for attending.
To start sharing my screen. It's in screen number 2.
Armando,
then my presentation, right?
Yes, I tried to interpret you as much as I can, but would you like to add anything to your past works? Would you like to tell us about yourself a bit more?
Well, I can tell about that at the end of my presentation.
Yeah, okay. You may begin whenever you want.
All right, thank you again, everyone. And on today's talk, I will be speaking about my own work on how we could search for life on Mars, and with a few starting considerations. You have seen now in the last day these wonderful images of Mars, the new Perseverance rovers and the Ingenuity rotorcraft. And we tend to forget how expensive these missions are. This particular mission, the Mars 2020 mission, it costed a little bit more than $2.9 billion, which translated to Turkish lira is about 24 billion Turkish lira. And in the currency we use here in Spain is about 2.4 billion euros. And I don't know you, but at least me, I have a hard time trying to figure out how much money is that. And so for me, it's always helpful, for example, to use units of measures that I can understand. In this case, I will translate how much money is that in the terms of what my car costs, which is about 14,000 euros, and try to figure out how much money that the one you spent in March 2020 would allow you to buy the type of car that I bought. And if you make the calculations, you can easily buy more than 164,000 of the cars that I have, plus one tire. And I still have a hard time in order to figure out how much 164,000 cars are. So, for example, if you calculate the length of my car, multiplied by the number of cars you can buy with that money, you will get to at a distance of about a little bit more than 715,000 kilometers. And that allows you to put in contact, because with that, you can easily get to the moon, for example. So that's the amount of money you need to spend in any of these missions that are being sent to Mars. But even if you have the money of sending your own rover to Mars, or to come up with your own mission to Mars, that's not enough, because it's not easy to do that. If you consider, for example, this picture is the crater left behind by the Schiaparelli entering and descent module, which was the proof of concept of the ExoMars mission, which crashed into Mars a few years ago. And that brought us to mind that almost 1/3 of the missions that had been sent to Mars have been unsuccessful. Most of the things that we send to Mars either crash into the planet, or just keep going away, or skip into the atmosphere, etc. And one of the most known examples of this was the Mars Climate Orbiter. The Mars Climate Orbiter was supposed to get into the atmosphere of Mars in order to study its atmosphere. But here you can see the planned trajectory of the mission that was supposed to go about 226 kilometers on top of the Mars in order to get into the insertion pass into Mars. But this is the actual trajectory that it took much closer to the planet, and that happened because in this case, the software that calculated the thrust that you needed to power the spaceship in order to go into the insertion trajectory was calculated in feet per second, and that got input in another software of NASA, which took that as meters per second. And so that caused that up today, we don't know what happened to this mission. Some say that it skipped entirely the planet, who knows where it is now, who might be crashing into it. So it's not just having the money, it's having the technology and experience in order to go to Mars. So that's why it is interesting to consider that on Earth we have a number of planetary analog that allows you to study on Earth what would you need to develop and to do in order to find life on Mars. For example, one example is the Canadian Arctic. There's a number of conditions, particularly with a low nutrient and high UV radiation and cold. A similar situation you will find in Greenland. Here in Spain, we have Rio Tinto, which we think it may be an analog of the type of water you have in the ancient history of Mars. A similar case you will find in the case of the dry valleys of Antarctica. And the subject that we'll study will be covering today is the Atacama Desert in northern Chile. Now, the Atacama Desert in northern Chile is not cold, but it is far the driest place on Earth. And so that's why it is interesting to study as an analog model of Mars. So, to put in context how dry the Atacama is, let's consider the climate there in Istanbul. And you get to see that last day, you will get to see a humidity up to 74%, which is translated to water activity, and we'll talk about a little bit further in the presentation about what water activity means for life, which translates to water activity of 0.74. And the UV index here is 0 out of 10, which is the typical scale in which you measure when you have to find protection in case of the UV radiation is too high. So, for example, yesterday in Istanbul, you didn't need to find any protection. But compare that to the place where I was born, which is an abandoned natural mining town in the high periodic corridor of Atacama, and you get to see that last day, water activity dropped up to 16%, which translated to water activity is only 0.16, and the UV index was extreme, meaning that probably that day it went beyond 4 to 15. You will have a hard time spending a noon time here without sun protection, for example. So we have been speaking about water activity. Water activity, in particular, is a way to calculate how water is available for life processes, and it's in the formal definitions is the partial vapor pressure in a system compared to the standard vapor pressure of water. In practical terms, water activity is simply calculated by the relative humidity at equilibrium of any system and divided by 100. So in this scale, for example, water activity of 1.0 is when you have relative humidity of 100%. So the interesting thing here is that most life on Earth, including bacteria, when confronted with water activity of 0.9, will either perish or go into some way of stasis mode. That is a waiting mode in order to wait for better water conditions around. Only a few yeasts are able to still reproduce a water activity of 0.6. But the interesting thing here is that in the Atacama, in particular in the high periodic corridor of Atacama, I know places where relative humidity, that's water activity, goes into 0. That typically happen in the afternoon hours, 3-4 p.m. in the afternoon, and we have brought a number of sensors there, and they all measure the same. That is that they are not able to measure any water in the atmosphere here. So why explain that Atacama is so dry? Well, there's a number of co-occurring factors that explain that. First is that when you consider the masses of air that are coming from the Atlantic Ocean and then meet Argentina and Bolivia, when they confront the Andes Mountain here, as they rise up, all the water that masses of air are bringing, they are all being left at this side of the Andes. And when they go into the other side of the Andes, they come out really dry, creating this rain shadow effect, which is not only happens in the Andes Mountain, but here is quite marked on why the water is all deposited in the west side of the Andes, while in the east side of the Andes, while in the west side of the Andes, where the Atacama Desert is located, that masses of air are coming there very dry. A similar factor happens in global terms, when the masses of air that are coming, that are happening, and then bringing all the water and rain in the equator, when that happened, all those masses of air cool down and rise up and then come back again in a big circle at the same latitude, more or less, where the Atacama is located. So you also have an input of very dry air coming from above. And in addition, the Humboldt current that goes all across, way up into South America, that is coming from the South Pole, from Antarctica. The water is, as you can imagine, is very cold. So you have little evaporation of water, and the little evaporation of water there, you have a few clouds, mostly fogs, that when they confront the coastal range of the Atacama, so none of the little water evaporation here comes into the Atacama. So these three factors explain why you will find some of the driest places on Earth in the Atacama Desert in northern Chile. And also here you have to consider that if you look at on tectonic movement of plates in time, you see after 150 million years, those climatic conditions more or less remain the same in case of the Atacama Desert, because the South American plate moved only little since that age. So the Atacama is not only the driest Atacama desert on Earth, it's also the oldest desert on Earth. That's explain why you see places like this, like Yungay. Yungay is a site which is located in the middle of the hyperarid called Atacama Desert, and we'll start talking about this site because it's in this place that in 2000 NASA and other colleagues from different institutions around the world came to this site and tried to understand it in terms of what you can find about life in such a dry place. I know that because I was there at that time just as an observer. But one of the things that caught my attention, that among other things, they brought this gas chromatographer, which is the backup instrument of the Viking mission. It's actually the mass spectrometer that was used by the gas exchange experiment that was undertaken by the Viking 1 in 1976, and also by Viking 2. And so what they found, and these researchers in 2003, that was published in Science, and they proposed that salts organic that were found only at trace levels, that you could only find really, really low numbers of cultural bacteria. They could not recover any DNA from its salts, although that changed in time in the next years. And when several of the experiments undertaken by Viking, by using salts of Atacama, gave more or less the same response. So that was instrumental, because for the first time, it was proposed that Atacama could be a proper mass analog model. And so since then, many research teams from all around the world are coming every year into Atacama in order to understand what the Atacama can tell us in order to find life on Mars. So in my case, NASA proposed that in order to find evidence of life on Mars, the first pass that would be valid to take that approach will try to find where is the water on Mars. If we find the evidence of water on Mars, since we know that water is required for all life on Earth as we know it, if you follow the places of where the water may be on Mars, then you could, as a next step, try to find evidence of habitability and maybe biosignatures, right? So I use the same approach, but in this case, in order to explore the Atacama. So for that, I started in the coastal range of the Atacama, because there the relative humidity is a little bit higher, and then they start moving inland in order to register what happens as you advance inland in the relationship between water and water availability and life. So, for example, in the coastal range of the Atacama, you could see in some of the cliffs in that area, you will see that black bandings, and those black bandings are actually a type of biofilms. They are able to survive here where water activity is quite high. It depends on the time of the day. it will change between 0.7 and 0.9. And that allows you to have a very complex biofilm there, composed of several photosynthetic pitches and also a number of heterotrophic bacteria and archaea and fungi as well. So if you look into the species you can find there, you can find representative of the 3 domains of life: archaea, bacteria, and fungi. We have been able to grow a number of these species there as well in the lab, which is not always easy in order to try to find the proper media and growing condition, temperature, etc. In order to grow species that in principle you don't know they are there, but we have been able to do that. And also in the coastal, in the same site, in the cliff of the coastal range, there's a different phenomena where you can find caves. This actually started in an expedition in 2009, where NASA was testing how you can use infrared technology in order to get to set from a distance where you have cave entrances that later was applied in the case of Mars. But in my case, I went into one of the caves. I tried to see if I could find a photosynthetic life. And why that? Because photosynthetic life was one of the first type of life form that evolved on Earth. So if that same happened on Mars, you still might find it inside cave, well protected from the harsh outside conditions in the case of Mars. Here, the water activity drops a little bit in the lower term, in the lower side from 0.59, but also get quite high up to 0.889, explaining why you still are able to have those biofilm of in this case of a particular microalgae, which is able to grow in this place. Is a type of microalgae from the genus Cnidium, which is able to grow with very low amount of light. That it is 0.06% of the amount of light you can record outside. And so this is an interesting phenomena in order to find photosynthetic life inside caves. Another caves in the coastal range, the water activity here this is a little bit, just a few meters inland, and that is enough for water activity to drop down to 0.14 in the driest hours of the day. But again, you get quite high up to 0.9, and there you can find that you can find photosynthetic life on top of the spiderweb that covered the cave entrance, not covering the rock of the cave, but covering the spiderweb. And why is that? Well, we found that the spiderweb silk is quite hygroscopic. So if you consider the fogs that I just talked about, that are coming out and confronting the coastal range, those spider webs are able to trap the water from those fogs and provide a source of water for this microalgae in particular, which is a new species of Dunaliella, and actually is the only one member of a genus of 25 species that is able to grow outside the water environment, and curiously, in this case, on top of the spider web. So in order to test our hypothesis, we went back to this cave, and not at noon as the picture I just showed, but we came back at 4 a.m. in the morning, and quite clearly we could see that those same spider web were actually covered with many droplets of water. Why these caves in this area are interesting in the case of Mars? Well, because as I said, maybe in the case of Mars, you could find not maybe extant life, but maybe the biosignatures of past microorganisms, photosynthetic microorganisms. And the idea here, they may be well protected inside the cave, so not to be affected by the harsh UV radiation environment on Mars, but close enough yet to that same cave entrance, so have a little amount of light in the range of between 300 and 700 nanometers, which is the range that where photosynthesis take place in order to do that process. So actually, I take the opportunity to tell that we have just accepted a manuscript in Nature Astronomy, in where we're proposing what are the steps we should take in order to explore caves, for example, on Mars and on the moons. In the case of the moon, in order to maybe use it as habitat for future human astronauts, but in the case of Mars, in order how we will explore the caves on Mars, of which we know there are many caves on Mars, in order to find potential signature that, given that they are inside caves, they may be better protected from the environment, and that's to be investigated. Now, if you move then a couple of kilometers inland in the coastal range, the main source of water here still are the fogs, and in a few sites of the coastal range of Atacama, you will find these quartz rocks outcrops. And in this site, if you pick up between around 8 out of 10 of these quartzes, you pick them up, you will get to see that they are below, and then you will find these beautiful green biofilms. And these green biofilms, again, are composed by a number of different bacteria and photosynthetic bacteria. I'm showing another picture here. Water activity here, again, is quite low, depending on the time of the day, 0.16, but it gets quite high to up to 0.95%, again, because of the fog that covered these areas in the late afternoon hours and during the night. And so these photosynthetic species and also the heterotrophic species that are live associated to these species are able to live there because the quartz stone is quite transparent to sunlight, including the range appropriate for photosynthesis. So depending on the quartz stone, between 1% and 8% of the solar sunlight is able to get down to the bottom of the quartz stone, and that's explain why you have enough light for these species to survive. And we also find here that also it's very interesting that this quartz stone remain much colder than the environment during the nighttime hours. So the light plus the fogs plus the low temperatures during the day allows you to propose a model in which these fogs covered all these areas in the late afternoon hours and night. But given that the quartz stone are much colder than the environment, they are able to retain maybe more water or maybe retain it longer during the day in order to allow this biofilm to get a place below those quartz stone. And given that many of these species, particularly the cyanobacteria, are able to produce extracellular polymeric substances, which are rich matrix of sugars that are also quite hygroscopic, they maybe get the system going in order to trap more and more water and keep it more efficient during the day. And that allows you the quartz colonization and the biofilm we're observing here. If you inspect those biofilm, again, you find representative of the 3 domains of life of at least 7 different species belonging to the bacteria, the cyanobacteria, the archaea, and the microalgae. So this system are quite island of biodiversity in a much drier environment. And this is interesting because it tells you a little bit on how you should search for life from Mars again. Again, these are the latest pictures of Perseverance, for example, and it calls for, in my case, it caught my attention one of these rocks because this rock have this little void space, and maybe life, you will not find life in those void space inside exposed to the surface, but maybe if this rock have voids inside the rocks or below it, maybe those are the places you could find evidence of life. No, maybe not exactly there, but the general concept that you should look for protected environments that will tell you a lot. They may tell you a huge difference on where to search for life on Mars. Maybe you will find evidence of life here because you have a protected environment under the rock, or maybe in this space here, but you maybe not find anything in here, right? Which is the same case we are finding in Atacama. Imagine this is a quartz stone. You will be able to find life below the quartz stone, but nothing or little less, much less than life from here in the soil itself. So one important lesson that in this very extreme environment, the importance of microhabitat is very crucial in order to understand how and where to search for life. So moving inland in Atacama, we come out again to Jangai. As you remember, Jangai was described by NASA as one of the first analog model of Mars on Earth in the Atacama in particular. Here, water activity dropped down to zero in the afternoon hours, as I said, but also it's quite high in the early evening, 0.86. And NASA proposed that maybe this was one place where you could find the dry limit for life on Earth. That is, a place so dry that nothing was able to survive in it. But I knew that was not the case. And why? Because I was born and raised in the Atacama. So I knew that it was very usual that when you drive through into the road that brought you to Yanga, you get to see those heavy fogs. And as I told you before, fogs are one of the main sources of water for life in the Atacama. And that is evident if you go into Yanga and you get to see those little rocks here. And those rocks are actually made of almost pure sodium chloride, salt, the common salt you use for dinner, for example. So in this halite, you'll get to see those gray bandings, and these gray bandings are actually a type of cyanobacteria, which is Crocodidiopsis. And I'm enhancing the pictures here in order you can see the cells. You can easily extract them from those halite just by dissolving in water, telling you that actually you have enough water for life even at the surface. Although they are inside those crystals in an endolithic mode of life, meaning that they are living inside the rocks, you still can find life here, proving that it wasn't the right place that it was supposed to be. So it took us a few years in order to look for the really dry places of the hyperotic color of the Atacama, and then we found Maria Elena South. The name is given by because it's south of a nitrate mining town that is still active, which is Maria Elena. And actually, this is the driest place of the Atacama and the driest place on Earth, because water activity again drops down to 0 in the afternoon hours, but it doesn't get higher than 0.58. And so this is actually the one of the places that you could find whether you could find here the dry limit for life on Earth, and that actually we're looking for places like that when we found Maria Elena. So we inspected in the soils, and we set a number of sensors in the relative humidity sensor in order again to assess water activity, the amount of water available for life in the soils of Maria Elena. And we found that if you measure the relative humidity at the surface, you get to see, as I said, that during the day, they will get really, really low, up to 0 in some cases, as I'm showing here, and it will go up into the night, up to, as I said, almost 0.6% of water activity, 60% relative humidity, right? But if you go 5 centimeters down, and then if you go 20 centimeters down, and if you go 50 or 80 centimeters down, you get to see that the water activity below is quite low. Actually, at 20 centimeters down, the relative humidity, you have only 7%, which is equal to a water acidity of 0.07. And that was interesting because it was the first time that someone was able to find a place on Earth that was as dry as that. And actually, that place is as dry as Mars. And why we know that? Because here in where I work, in the Center of Astrobiology, Spain, we have provided the meteorological instrument for Curiosity and Perseverance, which are these little instruments here that are able to measure temperature and relative humidity. And so with that, we know that if you measure relative humidity at Gale Crater, which is the case of Curiosity, you get to see that the lowest relative humidity that was measured a few years ago here are around the same range of the relative humidity measured at Maria Elena. So with Maria Elena, we have found a place that, depending on the time of the day, is as dry or even drier than Mars. So the question is this a place that was so dry that nothing is able to survive in it? And we thought it was. But after taking all the proper measures and control, we found out that depending on the depth of the soil profile, you will find a number of different bacteria, very low diversity, only species of Streptomyces, Bacillus or Dermatophilus, but life is there. Maybe it's stasis mode, waiting for better conditions. We still don't know. We don't know exactly the levels of metabolic activity that these species have under such extreme dry conditions. But we are investigating now. And even we have hints that this species, which is Dermatophilus, is maybe the primary producer, because Dermatophilus is known for being able to live from the reduction of manganese, which is present in this soil. So this is maybe the primary producer, and maybe Streptomyces and Bacillus, and maybe a couple of other species we haven't been able to home yet here, are able to survive from that primary producer. So the next question we ask is, well, how life get into the Atacama, and how you explain the presence of this bacteria in the soils of Atacama? And I know more. I knew more or less that the answer was related to the wind, because in the Atacama, wind is a constant. Every afternoon, after 4 or 5 p.m. in the afternoon, you will have winds that are coming from the Pacific Ocean, from the south, and then go inland into the Atacama. So we set a number of transit across these wind corridors, and we put different plates with different gross media, and also empty plates in order to assess the amount of dust that this wind was transporting around. And we found, as I knew, that the wind speed in the afternoon were much higher than in the morning, and also that you could trap a number of enough amount of dust in each of these places. Although I'm showing here an amount of dust in gram per meter square, actually that translates only to a few milligrams of dust per plate. So I wasn't sure I would be able to find any microorganisms associated to that dust. But to my huge surprise, I found about 80, if not 90 different species that were able to be extracted from the dust. I mean, they were using the dust as a way of transport in order to move across the Atacama. We could also follow the path of movement for the different species. For example, these 4 species here were able to traverse the entire Atacama. So because we will find it inside 1, 2, and 3, which is comprised about 100 kilometers here. Many of the species we just find it only close to the coastal range, and a few here in the site that were farther inland. We were also able to model the wind speed and trajectories, and we understood that the transport process took place mostly in the night, in the afternoon hours, late evening hours, and during the night. And with the wind speed and that time of the day, dust and the microorganism that was transported by that dust were able to reach and even grow across the entire Atacama, which is about a 300-kilometer range here, in a matter of a few hours only. And that happened during the night. And given that it happened during the night also, that is the time of the day where you don't have any UV radiation, and also, if you remember, where the relative humidity in general gets higher. So life will have the best of conditions in order to move this harsh atmosphere during the night, because of the lesser UV radiation, the high relative humidity, in order to move in a viable form across the Atacama. That happened during the night, and during the day there was a lesser movement of this dust, but only in the areas around. So we can understand that even in the Atacama, with being so dry and with so much UV radiation, life could use the dust as a transport if you fly around during the night hours. And that is interesting because, again, in the case of Mars, we know that dust storms are quite common and even reach a planetary scale. So did that happen in the past of Mars, in the ancient Mars? Maybe life may have originated in better environment like this one. This is what we think Gale crater looked like in the past of Mars. Maybe life may have arisen in this place and then transported by the wind to places somewhere else. So that is an interesting mechanism we want to explore, and we are thinking about an instrument in order to, in the next mission, to trap the Martian dust. First, in order to characterize it, in order to know what it's made of, and then, if hopefully, maybe it's transporting biosignatures. And that's the way. That's the lessons that you learn when doing this kind of experiment in Mars analog models. And now, following in the findings of the Atacama, then changes came that no one expected. These lagoons was served in 2015, where we were traveling to the Maria Elena South. We first would see one of these lagoons, then another one, and then another one, and then we stopped in order to take a few videos and pictures because we didn't think that this could have a natural origin. We saw that given the high mining activity all around Atacama, this was the leftovers of mining activities around, and so we didn't give it much thought and just keep driving. That happened in 2015. As you may remember, I said that it doesn't rain in the Atacama. That the rains are very low or non-existent. Actually, there is place in the Atacama in the hyperarctic core There is a small city here that is called Kiyawa, and Kiyawa, it has a meteorological record of 450 something years, and they have not recorded a single rain event. So that is how dry it is, but it's not uniformly dry. As I said, most of the time clouds are stopped with the water that are bringing coming because they confront the Andes Mountains that are located there. But most of summers in South America, those clouds are able to traverse the Andes Mountains, and then you have some rains at the other sides of the Andes, but only at the foothills. So you have in the foothill of the Andes an ecosystem like that, where you have a different number of animals and plants, etc., which is quite different of what is happening in the high periodic core. This is those are the places that have been shown to you. But what happened in 2015, then we understood that, and that happened also in 2017 was quite different, because in this case, clouds came from the wrong direction, so to speak. They came from the Pacific Ocean, and so we have very unusual rain in all this area that had in a climatic event that had never seen before. I mean, we have never seen clouds coming from the Pacific Ocean and bringing rains, as it happened in 2015 and then happened again in 2017. So this created, again, rain lagoons in Yungay, in a place where no one has ever seen water around. And then we understood, and this is the work of others, that they understood that in the 2015 and 2017 event, what happened is that we think that due to climate change, the temperature of the water of the Pacific are getting abnormally higher. Higher temperatures, higher evaporation, higher amount of clouds, and then you have the clouds that go inland into the Atacama and in a place where it hasn't rained for a million of years. So we've seen that happen. And actually, there is a statistics, again, this is the work of others, that is showing that in the last decade, it has been raining as never before in Atacama. You can see it in the blue area where it has been raining more, and in the brown colors where it has been raining less or the same than before. And you can see actually here that Yungay, the place where I just showed one of these lagoons, actually is in one of these areas where it has been raining more. Not so in the case of Maria Elena, which is still remain one of the driest sites of Atacama, to our fortune, by the way. So you would think that in this environment, water will be welcome like manna from heaven. You know, water so much needed for life. But when we inspect the water by microscopy, we were expecting to find all type of little things moving that way and the other, but we could not see anything. And actually, I had to filter this water in order to extract some DNA in order to find just 4 different bacterial species, the one that I'm showing there, which was coherent with this 4 morphotype of a species with the type of cell we could see in those same filter water. And we were able to grow one of them. And the question is, well, we knew the species composition of this soil, including the Cerabacteria that I showed inside the hallux here. And we found that if you compare the species in this lagoon compared with the species in the soils that were dissolved by this water, 85% of the remaining species just completely vanished. And we think that happened because of the very well-known effect, which is called osmotic shock. These species, we think, are so well adapted in order to trap, keep, and use the water when confronted with such amount of water in such short amount of time. They simply don't know how to deal with it and just simply burst with water. And we know that because we can find some of the molecular remnants of that. For example, pristine and phytans are typically the lipids that you can find in the membrane of cyanobacteria. Remember the cyanobacteria I showed that were found inside the hallux? So we are not seeing the cells here. We could never see a cyanobacteria in the water of this lagoon, but we could only see the biosignature left behind by the lipids of the cells. But also we could see other biosignature like the 16-meta-heptadecanoic acid, which is the typically biosignature of living cells, which are the cells that are actually living in those lagoons. So you could see a mixture of biosignatures from the species that were burst from osmotic shock, as well as the biosignatures left of those 4 species that were able to live in those lagoons now. So here, incredibly, water caused the local extinction of life. which makes sense in terms of the evolutionary terms on how life has adapted to this environment and then confronted with a sudden change. This also caused that in another side of Yangai, it took only about 30 centimeters to go into the soil and then find water again in never seen before in the history of Yangai, which has been researched for the last 25 years. And that actually water is contained in a smectite composite type of clays that are holding this water here with a water activity you can see here 0.78, and that compared with the water activity on the surface, that much higher, and that explains that when you have clays and you have water, you have fine life. And here you are seeing all the species that you find by direct DNA extraction of these soils, a number of different species, bacteria, and also archaea here. We also were able to grow some of these species as well here. We could see also too that these species, we could see it in the soil by cybergreen staining, and you could see when you have these little clouds around the cells when you are staining with cybergreen in soil, that tells you on the presence of exopolysaccharides. And when you have a group of cells surrounded with exopolysaccharides, it tells you that there maybe you have not only one but several species surrounded by the exopolysaccharide. That tells you that maybe some functional collaboration between those species there. And also, if you use CTC staining, which allow you to assess the metabolic level of the bacteria, you could see that the bacteria actually were metabolically active. And so this findings were interesting in the terms of the environment again, because as you remember, in the case of the soils of Maria Elena, here I'm showing the temperature profile of Yangai. Surface temperatures quite high during the night, quite low during the night. At 20 centimeters and then at 40 centimeters, where the clay layer is located, the temperature is almost constant in time, around 70%. In the case of relative humidity, again, relative humidity will go up into the night and then go really low during the day, but it was the opposite in the case of Maria Elena. Here, the water activity was quite high underground, where the clay layer is located with a water activity, as I said, 0.78, and that is enough for life to survive there, and not a few species as I showed. And so this is interesting again, because this is what proved that, for example, missions like ExoMars that are going to drill to the soil and going up to 2 meters of depth actually are looking in the proper place. I mean, if you are going to look for life on Mars, as we have seen in the camera, it has to be a protected place where the local conditions command that you have enough water for life in order to survive there. So look at the general trends. If we retrace our path into the Atacama here. In the coastal cliffs of the Atacama, you can find life that is exposed, yet still exposed to the environment, because water activity is quite high, between 0.7 and 0.9. So life is still able to be exposed to the sun, so to speak, and live in the epidictic mode way, right, on top of rocks. But if you go just a few kilometers inland in the coastal hills, life start taking some kind of protected environment in the epolytic mode way, a way of life that is below rocks in this case, where water activity, as you can see, is as high as the other case, but the lower end is much lower, 0.16, which is a typical day in the Atacama. If you go into Yangai, the water activity in the high range drop a little bit to 0.8, but then you get really low during the day at the lower range, just 0. And if you go into Maria Elena at 20 centimeters of depth, that is the driest place, and then a water activity of 0.1. And then the only way you can survive there, in order to survive the dry environment and the high UV radiation, is to bury yourself into the soil in order to at least be able to survive around. And another way to see also this is that if you want to understand how life may have evolved as Mars got drier and colder in time, I can tell you what are the type of mode of life and also the type in the place of the Atacama you could find it. So, for example, during the Noachian and maybe part of the Sperian, you could find life exposed in the Epilitic or maybe in Politic as Mars was getting drier. Then you have to take further step in order to be better protected in the transitions from the Sperian to the Amazonian in the Indolitic way of life that is getting into rocks. But if you want to find life now on Mars or the evidence left behind, probably the best place would be go into the depths. And also I can tell you that understanding that, I can tell you precisely where to go in the Atacama in order where you want to understand what happened in the Noachian or the Sperian or the Amazonian epochs of Mars in order to find the proper model for each of those epochs. And again, the main lesson here is that if you go, I cannot tell you what are you going to find on Mars, but I can tell you where to look for it. And as I said, the best place here in order to find life is go underground, and that's why, for example, missions like SMRs and the future missions are considering going into those places, which is not easy in order to get those samples in order to analyze them, in order to see if you have a source of water and a better protected environment that where you can find extra life, maybe, but at least the biosignatures left behind that still can be protected down there. And I will finish my presentation just by giving you a hint of the place we're studying now. This is a place called Redstone, which is, given my experience, one of the closest analog models of Mars on the Atacama that we have discovered up to now. A place rich in hematite, a place where we have found a number of different microorganisms where we don't even have an idea of what they are. We can just tell they are bacteria, and that's it. And so we're now studying in this place now, and the critical thing here, we are studying with the actual instrument on the mission that are going to be sent by ExoMars, and the instrument that are Perseverance and Curiosity using Mars in order to find out what real instrument would find in places like this one. And with this, I end my presentation. Of course, I talk about a lot about research. You can read all of my papers that are freely downloadable from my Research Gateway website. You can find all the papers here. If you have any doubts on any of the things that I just told you, and please, I will welcome all your questions, and thank you again for the organizer for inviting me.
Okay, so let's move on with your Q&A session then. Okay, our first question is, any idea of the time scale for dust-transported bacteria to sustain a colony? Can the water vapor transport above Jezero crater to be sufficient resource? Or do we expect to find fossils of bacteria?
Yeah, that's an interesting question, because in the case of the work we did in the Atacama, as I said, life was able to move across the Atacama in a range of about 300 kilometers in a matter of hours. So it could easily traverse the Jezero crater, for example. And now I also show that the relative humidity in the case of Jezero, it can go relatively high, depends on the time of the day. So as I said, if you travel around during the night where you have a higher relative humidity, you could still be viable in the transport process. Not so during the day. During the day, it will be too dry. You have, again, the sun, you have UV radiation, including the UVC that you have on Mars that we don't have, luckily for us, on Earth. So you could, in principle, move across Mars during the night. And given that this is an ongoing process, and then you have global Mars dust storm every year, it's just a matter of statistics that maybe if life is around, it could move from place to place if you find the proper conditions. So that also applies to biosignatures. Maybe you have a place on Mars where you have ancient biosignatures, for example, the remnant of bacteria, that if you move it around in the proper time of the day, as I said, during the night, you could move those flight signatures from place to place. So that is an alternative, and that is why we want to study in close detail the Martian dust in order to see if that could be the case.
Okay, our next question is it possible to create a life under Martian conditions with the microorganisms that are adapted to conditions that we have in laboratories, in our laboratories? Can we start the evolution process with this?
Yes, and actually, there is already people that already proposed this. I think one of the first that proposed such a strategy was Chris McKay from NASA Ames Research Center, which is with the same investigator I showed you a picture, the one that first proposed that the Atacama Desert as a Mars analog model. And so he proposed that in order to terraform Mars, and that was a science paper, and actually it was the main image on that issue of the science paper, where he proposed that among other things you should consider in order to terraform Mars was to take those species from the harshest environment on Earth, like the Atacama Desert. Probably they by itself they would not be able to survive in the Atacama, but through genetic engineering you may enhance their ability in order, for example, to tolerate UV radiation, to tolerate extreme desiccation, in order to use those species of microorganisms to start the terraformation process. So actually there is people working on that, and they are testing those in genetically engineered cyanobacteria in Mars simulation chambers in order to measure how they are able to survive in actual or simulated Martian condition, thinking about where you could be able to start such a process.
Or can we create laboratories that are much more like Martian surface?
Oh yeah, of those there are many. Even here where I work, we have a Mars simulation chamber. It's about this big, more or less, the inner space. But I remember have seen a simulation chamber in Kennedy Space Center, which is the size of this room here. So yes, people already has those instruments in order to do those kind of experiments. The strategy of using a Mars simulation chamber is well known for a few years now already. That's the way you test those species.
Can we make the microorganisms we have on Earth adapt here with how can I say this, simulations, and then send to Mars, and maybe they'll survive? Is this possible? Is this ethical?
There is already people working on that, but the issue is that no one will allow you to take anything alive to Mars because we don't want to contaminate life with Earth with life on Earth, right? So that may happen maybe 100 years in the future or something like that, maybe 15 years in the future, if we never ever find any evidence of life on Mars. Maybe people will start thinking, well, how we can change Mars by bringing Earth or microorganisms we have evolved on Earth, using the different techniques in order to do that.
Also, I have a couple of questions from my notes. And what does a day look like working in the desert all day long and seeing all kinds of different animals and beings?
Actually, it's a very strange sensation because given that life is not evident in the Atacama, when you go, of course, for me, it's nothing new because, as I said, I was born and raised in the Atacama. But for people that have gone first, it's very amazing to be in a place which only seems entirely devoid of life, and you have to bring your own expert with you in order to tell you life is here, there, and there. And there's many interesting phenomena happening in the Atacama. For example, I know places in the Atacama that, given there is the relative humidity around is so low, and the air is so still in the morning hours, that you can talk with someone that is 200 meters away from you, and you can easily hear that person and keep a conversation. So
Wow.
phenomena happen in those places that are very, 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 very unusual. For example, I have a couple of colleagues that have not followed my advice to drink water, even if they don't feel like drinking water, because there is so much evapotranspiration from your body that you lose a lot of water just by being there. And so I ended up in 2 cases with colleagues in the hospital because they didn't drink as much as water. You typically in a day in the Atacama, you typically drink between, just by moving around, even more if you are digging a hole or something like that, 7 to 6 liters of water, and you don't even notice that you have drank those 6 or 7 liters of water because it's so dry. Remember, you are in an atmosphere where relative humidity is 0, so any water from your body is getting really taken from you very fast and strongly. And also, again, the same thing with UV radiation. If you don't use sunscreen, you will turn as black as you can imagine, just being exposed by 5 to 10 minutes there.
Like, it was really hard for me to imagine when I searched Atacama Desert, because Turkey, we have 3 seas, and it's very, like, dryness is rarely found, and we don't like it. So I could not imagine. It was so magical to me. It is really like what I would imagine Mars is.
Yeah, it is an amazing place, and actually, as I said, I know places that you can say this how Mars should like, or this is like standing on Mars, or other places where you can say this is like standing on the surface of the moon. And if you want to look at, give it a closer look, you can check my YouTube channel video, and you can find many of the videos I have uploaded there on different sides of the camera in order to have an idea of how they look like. And I always say that I know amazing places in the camera you could easily film the next Indiana Jones movie because they are so amazing.
Yes, the scenery is amazing wherever you look, and like, I haven't been there, but I've seen a lot of videos of it, and it's just really. Wow, it's amazing. And what would you say, what life form in Atacama Desert or other very dry places is the most exciting or weird one? Because I've seen
actually here I'm using just one model species, which is a type of cyanobacteria that I was able to isolate from the quartz biofilms. That cyanobacteria is from the genus Gliocampsopsis, and we have found, we have the preliminary evidence first, that is extremely tolerant to desiccation. And as you may remember, I showed this water activity chart where I said, well, if you go beyond water activity 0.6, most life and any metabolic activity stops. But I have pretty good evidence that in this case of this cyanobacterium, which I just consider a model representing all the types of bacteria that evolved to survive in Atacama, they may be showing still metabolic activity, some levels of metabolic activity at water activity of 0.2, which in a water activity range where nothing is supposed to happen. We have seen gene transcription. We have seen the biosynthetic routes still ongoing. So that's one thing we're inspecting in detail, and also the extreme UV tolerance. This species has a huge UV tolerance. We have tested that using a synchrotron. We have used the synchrotron that the Brazilian hubs in the city of Campinas, and there we have exposed it to the UV exposure that you would receive in the stratosphere for a year, and you will get to see that viability will drop only to 70%. But if you simulate, for example, the surface of a comet of an asteroid, where you mix those bacteria with just a 1 millimeter of silica, for example, the viability of those cells, again, exposed by the UV radiation of the sun at the level of the troposphere, the viability that doesn't drop nothing. I mean, the viability is still 100%. So trying to understand how this species are so tolerant to UV radiation, so tolerant to dehydration, it is very intriguing, and also tells you about the limits for life on Earth, and also what could you expect for the limit of life on Mars. I mean, if you're finding life that is so tolerant to those factors, dehydration and UV radiation, why would you not expect that life on Mars follow a similar path during its own evolution.
Yes, it's very
And
clear.
also to finish that question also, I found this, I call it the thing, just a thing, because I took this small video on the quartz rock on an even much drier place. And this video I have shown a number of different aspects and expert on the contaminant is not a bacteria, it's not a cyanobacteria, it's not an archaea. We don't know what the heck it is. we simply don't know what it is. You can see just a little blob with little things moving inside, and that's it. And no one until now has been able to tell me what it is. That is a fungi, is a bacteria. What they can say that it is not this or that, but no one has been able to tell me what exactly it is. So there's still a lot to be found around in Atacama. Again, remember, it's a desert that has been dry for the last 150 million years. Well into the age of the dinosaur, this place was already really dry.
Yes, very surprising actually. And we have one more question. What are the factors that are considered when choosing a location as an analog site for a planet like Mars?
Well, in the case of Mars, of course, the lack of water. Mars is really dry, as I told you. Ionizing and non-ionizing the radiation, because Mars doesn't have a ozone layer, because it doesn't have oxygen in the atmosphere. So all the UV radiation, including the very dangerous UVC, goes 100% fully into the ground. And since it has a magnetic field, it doesn't stop any of the ionizing radiation. So the main issues here are radiation and desiccation and cold temperature. So those are the you would say the three main factors. In the case of the Atacama, you don't have the cold condition, but you have UV radiation and you have desiccation. And if you go to the dry valleys of Antarctica, you have desiccation, but not as dry as the Atacama, but you have now the other factor, which is low temperatures. But it's interesting that in the case for life on Earth, cold it is not a problem. I mean, just think about polar bears, right? The cold conditions are not a problem for all the life that we know exists in the poles, for example. Desiccation it is, and radiation it is. Those are the factors. That's why we are focusing on those factors in order to try to understand what is happening on Mars in the terms of a Mars analog model on Earth.
Yes, thank you. Any comments on the chemical composition of the clay when you investigated in those dry environments?
Yes, in the case of the clays that I showed that are below the surface soils of Jangite, they are composed of smectite and aegite. And on smectite, it is interesting because these are the type of clay we're expecting the Perseverance is expected to find on Mars. And why clays are important? Because as you may remember, clays have this layer structure, layer after layer after layer, like a cake, right? So in between those layers, you can have water, and you can also have biosignatures. And actually, the clay environment is one of the best environments to look for ancient biosignatures because they are well protected between clay and clay layers. And that's why we're looking for clays on Mars. Not only that they are evidence of past ancient water environment, because clays only form when you have enough water for enough time, but also once you have the layer structure, you could store between them the different type of biosignature you could expect to find if you have ever if life was ever present in those water that are from those clays.
Wow.
And that's in the case of the Jangite clays. In the case of, I didn't say this, but in the case of the redstone, the last slide I showed, there we found another type of clay, which is vermiculite. And also vermiculite is very interesting because it's the type of clay that people expected to find in the landing site that where the ExoMars mission is going next year, with a more or less the same objective: try to find ancient biosignature that may be trapped in those clays. And who knows? If you have clays and you have water, as I've shown in the camera, maybe, who knows? No one can tell either yes or no. Maybe you can find life also, if it well deep enough hidden in the soil, right?
Yes, anything is possible when these extreme environments problems.
Anything is possible until you prove contrary. It's like being guilty or not, right?
Okay, I have one last question. I took a note of it.
Mhm.
So you said now climate change is also changing the environment of Atacama Desert and other dry places. What do you think in 5 or 10 years? How will this climate change play a part in the ecology and the living beings of these dry places?
Actually, that is a very pressing issue for us. Not only in order to understand how climate change is changing the ecology of the place, right? In order to see how the rains are affecting the ancient life forms of Atacama. Only also consider that it doesn't drain homogeneously, as I show in that map. It's draining more in places, some places, less in other places, and also in the kind of strategy you have to take in order to characterize those sites before they're, so to speak, ruined by rains, right? In order to rescue those bacterial species that are being annihilated by rain. So that's a right. That actually, I'm just finished writing a proposal in order to precisely that to do that as soon as possible, because also it's not only about trying to understand the limit of life on Earth, how you can understand the search for life on Mars. Consider that also, for example, one of the other things I'm doing, that we have found that many of these species that we are finding at Atacama are producing a number of different types of antibiotics. So, as you know, we have a huge problem, and that is the next problem after this pandemic. The next problem we're going to face, remember my words, it will be the rise in the number of bacteria that are hypertolerant to all the known antibiotics. And we haven't developed a new antibiotic in the last decade, not even one. So we need to search for new antibiotics soon. And the type of species you would be able to find really new novel antibiotics are the species, for example, the one you're finding in the soils of Atacama. So it's not only an issue of basic science, it's also about the potential application you could derive from those bacteria.
I understand. And some other question we took is, it's a really harsh environment, but do you think of hiring interns there?
Well, now I'm based in Spain, but I do have a colleague in a local university here that there may be a case for him.
We see.
Yes.
Just asking for a friend, of course.
Yeah.
Okay. Now, unfortunately, we have to end our session with you, but thank you so much. I really liked what you're working on, and I'm really inspired by it. As a biologist, I'm going to be a biologist, but not yet, but as a biologist, let's say. So thank you so much for coming today. We'd like to see you again in the future sometime. And thank you, and we'll move on with Dr. Chris. In about 7 minutes, after 7 minutes break. Thank you for joining us today.
Okay, thank you. Bye-bye.
Bye-bye.
Hello everyone, my name is Kardelen. I'm a member of Agenastro team and I will be moderating this session. You can ask your questions via YouTube chat box and I will try to ask your questions as much as our time allows. On our third session, we're welcoming Associate Professor Christopher Butch, who's currently Associate Professor at Nanjing University, Department of Biomedical Engineering. His topic for today's conference is One Among Million, the chemical space of nucleic acid-like molecules. Hello, Professor, how are you?
There we go. I'm doing well, thank you. How are you?
Thank you so much. If you're ready, you can start your presentation after you give us a brief talk about yourself.
Yeah, I've got that in here as the first couple of slides, so I'll just go ahead and get into it. Can people see my screens right now? Ah, great.
Yes,
okay.
You can see.
So my talk title is slightly different from the title in the it's not a syllabus handout. So I'm going to be talking about nucleic acids. I'm also going to talk a little bit about amino acids and phosphate. And the general linking story through my talk today is this idea of trying to understand the position of the chemistry of life within all possible chemistries. And so the talk is titled, One in a Million, Why Does Life Use the Chemistry That It Does? I am an associate professor in biomedical engineering at Nanjing University in the biomedical engineering department, obviously. And in my research, I use analytical chemistry, computational chemistry, cheminformatics, and machine learning. Lately, my work is leaning more toward designing new medicines and nanomedicines, but I also use these methods and models of biochemistry to study the origin and evolution of life. And I've been doing that for about 10 years now in various proportions of my time. So I started in science at Georgia Tech in chemical engineering with Charles Eckert and Charles Leota. And at Georgia Tech, I was a member of the Center for Chemical Evolution, which is a really nice NSF and NASA-funded group there who are interested in studying the chemical origins of life. And at that point in my career, I was much more of a bench chemist, although I was transitioning toward computational. And I looked at formation of glyoxylic acid and reactions of glyoxylic acid under prebiotic conditions as an entryway to making sugars and as an entry point into the citric acid cycle. The idea there being building off of a publication by Albert Eschenmoser, which is a beautiful read in Tetrahedron, I think in 2004, where he goes through a bunch of ideas about how glyoxylate could be a building block for prebiotic chemistry. And then we did some of the exploration in my doctoral work here. I then did my postdoctoral work at Tokyo Tech, where I worked with Professor Eric Smith and Professor Jim Cleaves. And the majority of the work I did there, and what I'm going to be talking about today, is kind of in this space of cheminformatics of prebiotic selection, trying to understand the characteristics of biomolecules that make them separate from other chemistries and might be the cause for which they were selected for incorporation into biochemistry as we know it. And then at NJU now, the portion of my work that is prebiotic chemistry oriented, we have a small project in my group looking at functional characteristics of alternate nucleic acid backbones, which is related to the work that I'm going to go through today. And then some work trying to use machine learning and different models of chemistry in order to infer kind of the process of protein evolution and metabolic evolution by unifying these models with databases like the PDB or different models of the geochemical record. Different people have different things that they mean when they talk about the origin of life or the chemistry of the origin of life. So I like to just give my kind of frame of thought as one of the first slides that I go into, just to kind of make clear exactly what I'm talking about when I'm perhaps being casual in my language. So this is just a picture of Mars. I don't study Mars specifically, but it's a good idea for what a lifeless Earth may have looked like, probably with some more water than we have on Mars. But in Martian environments, there's chemistry going on, and we know some of this from the different missions that have gone there. And we can infer other chemistries that are likely to go on outside of life from telescopic data, telespectrometry, these analyses of meteorites that have come to Earth. And what we know from all of these different data points is that the chemistry that can happen without life is still quite robust. And depending on the environment, depending on the abundance of different organic precursors, there's actually a quite wide variety of chemistries that could have occurred. And to my way of thinking, that means that it was likely to have occurred. It doesn't make sense to me personally that early chemistries would be very clean. And so then from that, there must be a selection process that occurs to go from this diverse, still abiotic chemistry to chemistry as we know it. And this is a difficult process to look at because the top-down methods, the ones that go backwards from what we know today, go through the bottleneck of the last universal common ancestor, which is widely believed to have occurred a fairly significant amount of time after the actual origin of life, and the geochemical record, which doesn't have a lot of detail to it. So we have to make these inferences. And comparing the chemistry that we have to all possible chemistries in trying to see what makes it unique is one way of making these inferences. So today I'm going to talk about 3 different stories of looking at modern chemistry and trying to identify the portions of it that are unique and why they would be then good candidates for incorporation in biochemistry. The first is just from the literature. It's a simpler example, but I like it because it creates a very straightforward kind of framing for why or how to do this type of analysis. And this is discussing why nature chose phosphates. The second portion of the talk is going to be about the natural amino acids. And then finally, I'm going to be talking about the chemical space of nucleic acids. So moving on to why did nature choose phosphate? This is a summary that is based largely off of this paper by Frank Westheimer. It's very short, very easy to read, about 2.5 pages, I think. And if you're interested in thinking about the chemistry of life and the origin of that chemistry, this is just an excellent paper to read and start to think about and use as a template for your own thoughts when you're thinking about why life might exist in the way that it does today. So in this paper, Westheimer points out that there's really 3 different roles of phosphate in biology. There's this structural feature element, there's a control element, and then there's its role in chemical reactions, which is probably the first thing that people think about when they think of biological phosphate, but it may not actually be the most important. As a structural feature, probably what most people are familiar with is as this linker in nucleic acid backbones. And so this is serving as a spacing element to join different nucleic acids. And one of the important features of this, there are several, But one of the important features is the fact that the phosphate backbone is negatively charged. And this means that it's going to provide a shielding effect against most common nucleophiles that could come in and break down the nucleic acids, because most of these under biological conditions are going to be negatively charged, things like hydroxide groups. And so the fact that the many nucleophiles are negatively charged and the phosphate is negatively charged means that these reactions are less likely to happen than if phosphate was a neutral backbone. The corollary of this is that it allows better control of when you want degradation to occur. So in enzymatic situations, what usually happens is you'll have a divalent metal cation that will come in and complex with the phosphate, and that creates a charge shielding effect under those conditions, much easier to degrade. And so this allows biology to kind of precisely control that. The third structural element here is the idea that this provides rigidity of the backbone. So nucleic acids, especially the nucleobases, are relatively low solubility. And having this negative charge there increases the interaction with water, makes them more soluble. And then it's also self-repulsive. So across the base pairing element, the phosphates repel, and within one backbone, the phosphates repel. And that actually increases the kind of difficulty of coiling DNA up and making it insoluble. And so there's entire biochemical processes that are dealing with controlling these repulsions when you're doing things like creating DNA coils and packing DNA. There's this role in phospholipid bilayers. This is another structural feature, somewhat similar to creating backbone rigidity. These phosphate head groups in phospholipid bilayers, they increase interaction with water, and they make these bilayers rigid, just like DNA. So they won't collapse in on themselves and form droplets. And this allows for formation of these aqueous oil interfaces that are so important to compartmentalization and keeps them from degrading spontaneously. As a control element, this is related to phospholipid bilayers, but when you have a neutrally charged molecule, it's much easier for that to just cross bilayers through diffusion. The example on the right is just glycerol with and without the phosphate. When you have phosphorylated it, the charge repulsions with the bilayer keep the chemical on one side or the other of the bilayer, whereas when it's neutrally charged, it can just pass through. So this allows an easy method for biology to control whether or not a given chemical is allowed into or out of a compartment. And then finally, for control elements, you can also control reactivity. So even when the phosphate is not directly involved in the reaction, phosphorylation can control whether or not a given chemical is going to bind with an enzyme. So you can use initial phosphorylation as a control step where you phosphorylate, which activates the substrate to bind with an enzyme, allowing a reaction to happen that wouldn't if the phosphate group wasn't there. And then finally, in chemical reactions, hydrolysis of polyphosphates. This is one of the most common chemistries that people associate with phosphate. This directly catalyzes nucleotide polymerization, and then indirectly through phosphoryl transfers can cause other reactions that are otherwise thermodynamically unfavorable to proceed. So there's these three crucial roles, and essentially the bottom line of Westheimer's paper is that there's no other residue that can obviously fulfill all of these roles. there's the structural role, the control role, and the reaction role. And you can break those out and you could potentially associate them with other chemistries, but there's not any obvious alternative to phosphate for one chemistry that can control all of them. So in a scenario where you're thinking about alternate chemistries of life, you would probably have to have an abundance of different chemical pathways to control each of these different roles. Whereas with phosphate, they're all sort of nicely packaged in one set of chemistry. And so the bottom line of this paper is that phosphate likely is an optimization, if not from the origin of life, shortly thereafter, that allows all of these different elements to be incorporated into one set of chemistry. Of course, that's not perfect. There's open questions. There's the question of when life started to use phosphate, as I just alluded to. No one knows whether or not the first life was phosphate-driven, or if it wasn't, what came before it, although there's a number of theories out there. There's an open question of how life overcame phosphate scarcity. So one of the issues with phosphate in the origin of life context is that it's both insoluble and unreactive under the typical natural conditions. There's no polyphosphate minerals. There's only one known pyrophosphate mineral, and there's limited geochemical production of the reactive forms. So these are kind of the arguments that people bring out against why phosphate would be in the earliest life, but then, of course, there's the Westheimer counterpoint of, well, what else would you use? And so altogether, this is a really nice way to consider a chemistry. There's no obvious alternative. There are all of these unique capabilities that phosphate has, and because of these, you can construct a fairly compelling argument for why life would choose phosphate, even if it wasn't the first choice. So with that kind of framework in mind, I'm going to talk about a paper that a group of collaborators at ELSI conducted a couple of years back. And the question here is whether or not the natural amino acids are a unique set based on their chemical properties. The paper is written up on the screen here, and then you've got photos of all the collaborators there in the author order. So the background here is that amino acids are not particularly complex. They're, of course, used as the building blocks for proteins, and all of the many structural and catalytic functions of proteins are derived from the properties of the amino acids. The different combinations of amino acids cause folding, and the different folds give proteins their function. Simple amino acids, both from among the set of 20 biologically utilized amino acids and other amino acids which have the scaffold shown on the right. To be an amino acid just means you have this amine and carboxylate group, usually spaced 2 apart, but not always. These have been found in meteorite samples and numerous experiments meant to simulate prebiotic chemistries. So it's not difficult to get to them, which means that why we use the set of 20 that we do is an interesting question in terms of the origin of life. Using just the chemical composition of the biological amino acids as your constraint, there's nearly 4,000 alternate amino acids that you can draw just by moving the atoms around. And so that means that whatever process caused the biologic amino acids to be synthesized and incorporated had a lot of other options to choose from just within that small set of chemistry. If you allow other atoms, if you allow more atoms, you can really create a quite large number of possibilities here, hundreds of thousands. So one of the interesting observations about the modern amino acid set is that the properties are highly refined in their ability to cover the distribution of size, solubility, and acidity or basicity of the residues. This is kind of an idea from protein engineering where people have recognized that these three properties seem to be the most important in being able to build protein to a given function. You need to be able to span different sizes to control your folds. You need to have different solubilities with different size and acidity properties to be able to control the folding of your protein and where you put functional elements, and you need these acid and base elements to control catalysis. And so the figure here is just a view of, this is from one of my collaborator, Melissa Alardo and Jim Cleaves, and I'm fairly certain Marcus and Steve Freeland were on this one as well, but the citation is just at the bottom there. What they're showing on the left here is just the positioning of the 20 biogenic amino acids in a box representing the chemical space defined by size, solubility, and then pKa. And what you can see is that they're relatively well distributed within there, fairly evenly. There's parts of that box that no amino acids are in just by the nature of the chemical constraints. And then the red and the blue sets are two non-biogenic sets that are better spanning in that space. And then on the right, that's just a plot of, I believe this was 2,000 alternate amino acids plotted within that space, which just shows that there are clusters of properties, and you cannot fill the entire box. But among the possible sets defined by those clusters, the biogenic set is really highly optimized in terms of its ability to span this chemical space. This is still about the same paper. In this work, they created 100 million random sets of 20 amino acids. Out of that 100 million, only about 6 million had better coverage in any one of those three properties. Only 20,000 had better coverage in two properties, and only 6 had better coverage in all three properties. And so what that shows is that obviously there was some selection process in this space for the amino acids to make sure that they spanned this as well. And that sort of also indirectly suggests that some of these other amino acids were around as options, because if you only had one option, then you couldn't select to make sure that the space spanning property was fulfilled. And we actually know this is true. The proteinogenic amino acids, those 20, are not the only amino acids that are in biology. There's a number of other ones that are used as metabolic intermediates, but aren't used as proteins. And this begins to suggest why those ones are eliminated from protein function. And then this is just getting into that a bit more. This set of 20 amino acids that we use, people who study this origin of protein coding believe that there's a lot of contingency here, meaning that the later orders dependent on the amino acids that came before them, and that they were encoded sequentially over time. There's an excellent paper by Trifonov in 2000 that goes through a lot of different models about this, and the thinking has evolved a bit since that paper, but this is still one of the best summaries within the literature. The general idea here is that simple amino acids that might have been geochemically produced would have been the ones that were incorporated into life at the earliest position. And then those ones would have learned the chemistry necessary to selectively include more complicated amino acids. But if a different set of early amino acids was selected, then the later ones may have been changed in order to accommodate the need to cover chemical space, as I was talking about. And you can also see this in some of the proteins that are encoded today. Not every protein has all 20 amino acids. Many lack one or more. The pie chart there is just showing ever decreasing numbers of amino acids. About 2/3 have all 20. And then another 20% have 19, another 10% or so have 18, and then it's just this exponential decay in the number of proteins with reduced amino acid counts. So our goal in this work was to understand chemically the possible trajectories that could have happened through this emergence order, these previous sets of amino acids that would get to the amino acids as we know them today. I'm using CAA here to mean canonical amino acids or the biogenic, proteinogenic amino acids. And XAAs are just all of the other amino acids. So the methodology here was to create random subsets of both canonical and non-canonical amino acids ranging from 3 to 20, and just compare the properties of the canonical amino acids to sets that include non-biogenic amino acids. And like the 2015 paper that I was discussing, the metric here is the range and the evenness of coverage in pKa, log P, and van der Waals volume. This is just the acidity, basicity, solubility, and size. And what we mean by range and evenness are the range is just the distance between the maximum and minimum property in each of the three categories, and the evenness is how well those properties are spanned. So you want, if you had 3 in a given property, you would ideally want 0, 1, 0.5. So the results when we did this comparison, we again did 100 million sets in each of the given numbers of amino acids. And what we found here was that the canonical amino acids were very good at creating sets that either tied in one of the 6 metrics we were using. So the maximal sets in this table here is the number of canonical sets that tie either the range, the even, or the evenness in one of those 3 properties. And on the right, what you can see is that there's basically an exponential decay in the number of non-canonical sets that are better than the canonical sets. So at 3 amino acids, there's a typo in this table. It's about 5% are better than sets of the canonical amino acids. At 4 amino acids, it drops to about 2%. And then out at 20 amino acids, as I said earlier, it's only 6 sets out of 100 million. And so this demonstrates that not only are these amino acids optimal in the modern context, they were also very highly performing in each of the previous set sizes, which would have preceded the canonical set. So no matter what the emergence order is, the canonical amino acids do a very good job of covering this chemical space. So what are some of the ones that do better? These are just the, I guess, 9 most common amino acids in better performing sets. And what you can see here is that there's these kind of oxidized versions of canonical amino acids. So you get these 2 oxidized prolines. And then you also get these amino acids that have multiple functional groups, which is not something that's terribly common in biology. So like the bottom right one, which is alcohol and an amine in the side chain. So you can see where these ones may have some properties that the 20 canonical amino acids don't. But for the most part, the non-canonical amino acids are just kind of trading around properties that the canonical set already has. So the takeaway from this portion of the talk is that the canonical amino acids, they're highly optimized to cover this chemical space for acidity, solubility, and volume. The decreasing prevalence of better sets is kind of indirect evidence, like I was talking about, that there's a experimental process that selects for the best amino acid sets. And then there's this open question of, well, I didn't show you connections between those sets. We didn't really look at emergence orders that are maximal throughout. So constructing those maximal trajectories and then constructing similar maximal trajectories through the XAAs would be another way to compare these that we have not yet done, and see whether or not those evolutionary trajectories look similar or if there's an advantage for the canonical amino acids. So moving on and treating RNA and DNA similarly, this is largely work from the paper here, which is myself, Jim Cleaves, Peter Berger, Jay Goodwin, and Marcus Maringer. And this is a similar look at nucleic acid space. So unlike proteins, there are only the 5 canonical nucleic acids for the most part. So we don't have the same kind of emergence trajectory, but we can look at their chemistry just within the space of all possible nucleic acids similarly. Nucleic acids, of course, are of interest because they carry our genetic material, and RNA specifically has been shown to be able to form structures and conduct catalysis. The core of protein synthesis in modern biology is the ribosome, which is an RNA catalyst. And because of this, many researchers believe in this idea of an RNA world that kind of predated the protein chemistry world that life as we know it inhabits. And the figure here is just adapted from Joyce, showing this idea that the Earth will form, you'll get water, that will create chemistry, and then from there you'll go into RNA, pre-RNA and RNA for your earliest function. And I just made the addition of this bottom set here to kind of underscore the idea that there would have been competitors to RNA in this scenario. So we have to answer the question of why, out of possible nucleic acid backbones, we would have selected the RNA that we did. So on the right here, these are just the different backbone scaffolds for RNA, DNA, and then the bottom box is a set of 9 backbones that are known to both form base pairing sequences with themselves and chimeric cross pairs with RNA. And that's important because the fact that these can both pair with themselves and pair with RNA means that there are other functional alternatives to RNA out there. These haven't been shown to be able to conduct catalysis, but the fact that they're pairing suggests they can make structures, and if you can make structure, you can probably make some type of catalyst. It's just a question of how rich that catalytic space will be. And of course, these haven't been studied systematically. These 9 were discovered based kind of on chemical intuition of how to build backbones that are similar to RNA and DNA. But all of these syntheses are very difficult. You have to form these very precisely controlled polymers. And so the study of this space hasn't been systematic because of that difficulty. So in this paper, we wanted to just see how the ribose backbone compares to likely alternatives. Our method here was to define the nucleic acid backbone as just as simply as we could. And so we said that it has to be able to connect to a base, and then it has to have 2 linker chemistries, which can be a hydroxyl or a carboxylate or an amine. And then we used a graph grammar method to just create all possible structures which fulfill those requirements. We limited these to 2 different sets, one with 3 to 7 carbons and 2 to 4 oxygens and as many hydrogens as you need to satisfy the structure. And then a nitrogen containing set with 1 fewer carbon. So just to explore the difference between the backbone sets when nitrogen is or is not allowed. And then in each of these formulas, B is just a given nucleobase. We didn't vary the nucleobase chemistry because that gets more complicated in terms of being able to pick out bases that are capable of pairing. So the first thing that we learned is that backbone space is really large. In the CHO set, there are about 86,000 possible backbones that can exist. And in the CHNO set, there's about 75,000. So in both sets together, it's about 160,000 different backbones. And of course, we limited a lot of the possible side chemistries. We limited a lot of the possible longer chemistries. So this is a subsample of what's out there. It's still an incredibly large space. The graphs here are just showing the number of possible alternatives, increases fairly linearly with increasing mass. And this is just a plot for every given backbone mass, as well as just the cumulative plot, adding everything below the given mass. And the reason you see that tail off is just because of a combinatoric edge. We limited the number of carbons, so above that 130 or so Dalton range, you stop seeing as many backbones as you otherwise would. So when you expand these out to stereochemicals, which of course you have to do to actually model a backbone, the 160,000 becomes closer to 1.2 million, which is a lot of potential backbones to investigate. Nowhere near that many have been done. If you look at just 5 carbon backbones, which of course is what ribose has, there's about 25,000 possible stereoisomers. And what we found is that most of the compounds here are going to be cyclic because, of course, that increases the stereochemistry of the backbone without adding another atom. The plot on the right here is a 2-dimensional embedding of the chemical space. The idea is that the closer together a given scaffold or a pair of scaffolds are, the more chemically similar they are. These are called Schuffenauer scaffolds, which means that only the core ring structure and its heteroatom substituents have been retained. So things like ribose and arabinose would both collapse to the tetrahydrofuran that you see in dark green at the bottom right. And the color intensity of each of these boxes corresponds to the abundance of that scaffold within chemical space. These 109 scaffolds that are in this 2D embedding represent about 45% of all the possible backbones. And so, of course, within each of those sets, there's a lot of variation still. There's a lot of stereochemistry, but this does begin to suggest a path for why RNA would be selected, and that is simply a stochastic process by which a large number of nucleobases were produced, and RNA was the one, or at least furanoses were the one that could be selected most easily from that set. I should go through one other element here. So at the top right, this plot was generated from a TS&E embedding. You can see the original embedding as the left-hand insert, and then the right-hand insert just shows how they were spread out to the grid. So the chemical relationships in the grid figure are a bit distorted, and you can see those better in that insert. This is the same set of compounds with another TS&E embedding with all of the scaffolds. There's about 1,200 total. And what you can see from this is that they tend to cluster into small regions of similar chemistry, and that there's an exponential decay in the number of scaffolds that have a given abundance within the system. And the furanose and hexanose type scaffolds are the two that are most abundant within this space, which is quite interesting in terms of that selection story. This is just a higher resolution version of the same plot, just with all of the scaffolds included. I'm going to try to zoom in on this just to give you a little bit better view. And you can see that the scaffolds tend to be pretty similar to their neighbors. You can see the furanose five carbon ring here on the left and the hexanose down below it. And the intermediates there, I think, are just dioxygen heterocycles. This is available in the supplement for the paper if you want a closer look at it. The linear scaffolds treated with a similar clustering metric don't show any kind of concentration. This is, again, another embedding, just showing all of the different clusters of linear scaffold. And there is no grouping in chemical space that has the same kind of depth of similarity as the cyclic structures, just because of the wider variance that these linear structures can have. And so the population is much more smooth throughout this embedding. So that's the amount of chemicals that are out there that can fulfill this requirement. But that's not actually answering the functional question of which are similar to RNA and DNA and thus could be expected to fulfill the same goals. The plot here is on the left column is just our CHO set, and the right-hand column is the CHON set. On the top, these are 3D similarity scores. So RNA can exist in two different helical forms, the A and the B form. These top plots are just the 3D similarity of every backbone in this set to these two forms of RNA. And the red elements in there are those known base pairing species that I showed you earlier. So what this shows is that you do not have to be chemically similar to RNA and DNA in this 3D space to be able to pair. It helps, they're clustered up there, but the PNA and the GNA are fairly different and are still able to pair, which means that there's probably a lot more base pairing backbones out there within this space. You can also see that by the method used, and of course this has some noise in it, you would expect RNA to have the highest score, but because of inaccuracies in the force field, it's not going to. You can actually see that there's a few species that the model thinks are even more like RNA than RNA, which is a little bit goofy. But what that means is just there's a number of scaffolds out there that are highly likely to be able to cross pair with RNA as backbone. And then the bottom plot here is the sum of those two scores from the top plot, plotted against synthetic accessibility, which is just a metric of how difficult the chemicals are to make. And so what you can see here is that GNA and PNA are the easiest on the right-hand sides of the graph. The locked nucleic acids are the most difficult, and RNA and DNA are somewhere in the middle with most compounds in this library. Again, suggesting that there's a lot of heretofore unidentified base pairing species out there. Very few of these species had been previously reported. So this is just a plot, pie chart of the novel non-stereochemical scaffolds for the CHON library and the CHO library versus the previously reported scaffolds. And so only a little under 1% have been reported, and those are biased toward the CHO compounds, since those are more chemically similar to RNA. So then we've got an ongoing effort in my group here to try to refine this further. We're using protonucleic acid builder, which I should have cited, but that's a recent publication out of the HUD and Williams Labs at Georgia Tech. You can find that paper fairly easily. To look at the ability of these backbones to actually form helical geometries, because that isn't covered in simple chemoinformatic analysis that I've discussed so far, and then use some molecular dynamic techniques to study the entropy of cross-pair, I'm sorry, the enthalpy of cross-pairing, so we can get an actual idea of what the distribution of other compounds are out there and better answer this question of how is RNA special within those. So the takeaway here is that there's been a lot of good work in recent years to think about how nature could have synthesized RNA prior to life. These are just a handful of papers. There have literally been hundreds that are worth reading. There hasn't been as much work on why it would have, and that's the question that I'm interested in and that we were trying to tease out with this work. And I think we begin to answer that with this idea that maybe it was a stochastic process, and it's just the fact that in a stochastic process, RNA-like scaffolds would have been one of the most abundant. So that there's not a kind of unifying bottom line across these. The main unifying takeaway is that in every respect, biochemistry does seem to be really heavily optimized in its monomers. And that suggests that there was a rich competition between monomers prior to the chemistry becoming set in stone. Because if there hadn't been, then there wouldn't have been any opportunity for that optimization to occur. So with that in mind, I want to make a quick plug about Nanjing University. So for anyone out there who's thinking about grad school, we are actively recruiting for international students. The university itself is number 5 or number 6 in China and in the top 120 in the world, depending on which set of rankings you're looking at. My department, biomedical engineering, is a new department in 2014. The focus is on nanotechnology and healthcare and things like protein, nano material composites, so lots of very heavy biochemical work, as well as then using nanotech in devices. Obviously, there's other work that goes on there, considering that I'm doing origin of life work in the department, but that's kind of just the broad picture. And me and my colleagues are actively recruiting international students. There's scholarships available for master's students, PhD students, and postdoctoral researchers that have a pretty decent salary associated with them and mean that you won't pay for anything in terms of tuition or housing while you're here. So if that's something any of you are interested in, my contact information is here. You can reach out to me. And other than that, I will be happy to answer any questions.
Thank you for your presentation, first of all. Now we have some few questions for you.
Okay.
First, Zainab asked, is there any chance to find a life without carbon, hydrogen, oxygen, or phosphates? Can non-biomolecules emerge life?
Well, I can't say there's not a chance, but I haven't seen a strong argument for it. One of the better ones, if that's an area you're interested in, is to go and look at the work of Lee Cronin. And he's got a system where it's entirely metals and metal oxides that actually feed off of a source of energy-rich molecules and build themselves up into complex structures. And when you take away the food molecule, they die. Whether or not you can argue that to be life, whether or not you think that's a basis for a different type of biochemistry is a philosophical argument. In terms of pure chemistry, that's one of the best examples, though. Of course, it's not information carrying like life as we know it. So if you restrict life to being able to replicate through something like genetics, then I haven't seen that.
Thank you. What do you think about other forms of DNA? Especially, I'm wondering about what you think about xDNA.
If you mean xDNA in terms of the extended DNA that was reported by the Cool Laboratory, I think in the early 2000s, those ones look more complicated to create just in terms of the structure of the bases. So I would be surprised to see those over the bases that we have, but it's certainly not impossible. If you mean XNA just in terms of like non-biogenic nucleic acids, I haven't seen a compelling story yet for why it has to be an RNA. I think there's a lot of other nucleic acid species that it could be. I think there's small pieces of evidence that suggest why it would be RNA, but I haven't seen anything overall that's really compelling to me.
Thank you. You talked about thousands of isomeric structures. Do you think there's a possibility for them to exist on other planets or galaxies?
Yes, I am certain some of those chemicals are out there in the world. If you make me answer which ones, then I can't. But everything we know about kind of chemistry in space and chemistry on planets, or well, not planets, but under conditions like on Titan, Saturn's moon, is that there is a lot of kind of unrestrained stochastic chemistry, which means that there's probably any scaffold that you can imagine within the atoms that are there is there somewhere. The difficult question is how do you concentrate it and how do you then get it to do something?
Okay, thank you so much. I want to thank you on behalf of my team for accepting our invitation and joining our conference.
Well, thank you for inviting me. It was a lot of fun.
Thank you so much. Goodbye. Now we will continue after half an hour. We are going to have a break. Thank you for listening.
Hi everyone again. Our next guest is Dr. Omer Markovitch. His research focuses on complexity in prebiotic evolution in man-made synthetic biological and chemical systems. He is also a research fellow of the Dutch Origin Center. Today, he'll be talking with us on the origin of life and prebiotic evolution. Hi Omer, welcome. How are you?
Hi, thank you. I'm well. Thank you for the invitation, and it looks like a very nice conference so far.
There must be a problem in
Can you
technical
hear me?
problem. Could you speak again, please?
Can you hear me if I speak louder?
I cannot hear you.
Let me connect with my...
Oh, okay. It was because of me. I'm sorry. Welcome. How are you?
So can you hear me now?
Yes, I can hear you now.
Okay, just a second, so we can disconnect this. Yes, I'm sorry. So thank you for this invitation, and thank you for organizing this. It looks like a very nice conference.
Thank you so much. How's the COVID going there? Where are you right now?
Right now I'm in Holland, in a place called Groningen. It's in the north of Holland. And I think overall we are beginning to see the end of the whole COVID and lockdown situation. But of course, it's going to take a while. I'm trying to make the most out of it. If I would take one thing that sort of one positive thing out of this whole Corona situation, maybe two, is that because of that, we are now able to have such virtual conferences all over. And it really, in my view, it really makes science much more accessible because now students can more easily join. They don't have to travel far. Their registration are almost non-existent. So there is some silver lining.
Yeah, definitely. In everywhere, in all countries, I can see a lot of people are now preparing conferences and treating their community by spreading the love for science and the curiosity for it. So, as you heard before, I try to interpret you as best as I can. Would you like to add anything about your past research and yourself? And
why don't I start by sharing my screen?
Of course.
I will say one word or another, but I think so. If you can see me now, or if you cannot, let me know.
Yes.
So my background is more in chemistry. At first, I was focusing more about hydrogen bonds in water, and then I transition into more systems chemistry, and from that, trying to think how can a chemical system be considered alive, replicate, and evolve. And I've been pretty much doing stuff around these concepts since then. So the ideas of replication and evolution and Darwinian evolution is something that we recognize, and it's very easy for us to understand in biological system, like in the cell, in animals, in us. But it is something that is more difficult to recognize if it's a big if it can happen in chemical systems. And maybe this helps me now to sort of take this to where I'm going to talk now, which, by the way, nice to see a nice representation of the blue marble all over the speakers. So the way I'm planning for this talk, I thought that it would be best if this is going to be more of a conceptual talk. I will try not to go into a lot of details. Sometimes I just can't help it. And this is going to be just about sort of an overview and introduction for the idea of prebiotic evolution. And then I will maybe finish by giving by showing how I'm actually approaching this problem in the lab.
It's perfect for our audience since we have so many different attending people from all ages and races. So you may start whenever you wish. I'll leave the floor to you now.
Thank you. So as I said, I decided to make this talk more simplified in many ways. At the end, of course, I will give you some references, how you can learn more and educate yourself more, and I'd be very happy to discuss this all over the conference platforms. But then when we are thinking about the origin of life, this is where I'm coming from, or the origin in general, it's a grand question, because we think about, the origin of the universe, and then the origin of stars and planets, and then one of them was Earth, but early Earth was dead. It did not contain life as we recognize it. And then somehow this transition from no life to life had to happen because we are here. But when you think about the origin and evolution, it doesn't stop there, because we want to know how we go from simple life into more and more complex life, into life as we know it. And then the question of the origin of life, how do we go from non-living Earth to a living Earth, so to speak, is still an open question. But maybe this is what makes it so exciting. And when we try to think about this, maybe understanding this question can help us understanding how life could have started, might have started. Maybe this will also help us understand, for example, when we go and we seek life or evidence of life elsewhere in the universe. And the problem is that, unfortunately, we only have one example, which is us, which is Earth. And we are actually finding it very hard to think of different life that is actually different than us. And we can consider this by looking at all these cool aliens. So they all come from different movies and the different sagas and different planets and all of this. But somehow, even though they are aliens and different, somehow they all share quite a lot of features with us, with humans. So they all have eyes and ears, so to speak, and they all have hands and legs. And luckily, also, some of them even speak English. But what if in the origin of life, the way life started, it actually started as something very, very different? So in this case, it's going to be something that's going to be very hard for us to decipher just by looking at biology today, at life today. And another problem or another challenge there is that for many parts in science, we actually do have some data, some evidence for the processes that happen. For example, when we think about Darwinian evolution, then we can find more and more fossils that can help us complete the picture, so to speak, and give us actual evidence of the process of Darwinian evolution. But the problem is that actually we don't have direct evidence for this exact moment of the origin of life. So we don't have a fossil, quote, a fossil, so to speak, that is this relic from the origin of life. We know quite a lot about what was the conditions on Earth when it was formed, right before there was life. And then once there was life, we have some evidence for it, and we can analyze and understand quite a lot of it. But then for this transition from no life to life, at the moment, we don't have any evidence. Maybe this is also what makes it so exciting, because you can come up with all sorts of crazy ideas and suggestions. And life is rather complex. So if we can understand how life might have started, maybe this can help us not just satisfy our basic curiosity, but maybe also help us better understand what is life. So when you think about the jump from a simple bacterium to the complexity of humans, actually, as big of a jump this is, it is largely believed that the jump from no life to this simple bacterium is far greater. So the jump from this random pool, if we do this thought experiment, the jump from this random pool of amino acids, the jump into a complexity jump into the simplest bacterium is far greater than the jump from this simple bacterium into humans. And this poses those challenges and difficulties. How can we actually address the origin of life? So we think that actually it is not so likely that the origin of life, this sort of jump, happened as one step, as a miraculous event. And we can understand that by looking at this following example. So imagine that we have this storage unit, this warehouse that has all the basic building blocks, all the material that you need to create a modern airplane, which is a rather complex thing. But even if it has all the right things there, do you think that this is a likely event that somehow a magical hurricane can pass through this warehouse and all the stuff somehow assemble together, stick together, and voila, we have an airplane? Well, of course, always there is a chance for this, but this chance is just vanishingly small that we can accept that it will never happen. And likewise, we are trying to understand this relevance for the origin of life. So if we continue now with the analogy of aviation, we actually recognize that the father of modern aviation is this Wright brother airplane. So we see that there are lots of similarities between them, but it's actually more different than similar, because the Wright brother airplane had the wings of paper and it had the simplest motor, maybe like of a motorbike or something, and it could barely fly. Nevertheless, it still flew, and still this is, and because of that, it triggered the age that is now we see the modern aviation and this modern jet. Actually, if we try to continue this analogy looking for like the origin of life, then what I'm looking for in the origin of life is something even more simple than the Wright brother airplane. You can think that I'm looking for this sort of paper airplane equivalent for the origin of life. Maybe now we stop for a second, and I will ask you, what do you think are the basic ingredients of life? And I'm more interested in focusing for minimal life rather than full-fledged complex life like we recognize it today. So why don't you take a moment and maybe also put your suggestions in the chat. What do you think are the basic ingredients for life? What makes life different than a rock? And when you think about this, I will challenge you even more, because the rock is like something very static. But then we can also think of something much more dynamic, like this geyser that just erupts and erupts, or this fire that is very dynamic, and unfortunately also fires can spread quite easily. So what is the difference between those fires and the rock? And then what is the difference between life, between a living system, let's say a cell, and the fire or the geyser? So you can think that the missing ingredient there to make it actually alive is not just the ability to spread, but it's the ability to spread in a very specific way, to replicate by transmitting your information, by copying your genetic code to the next generation. And this is something that a cell or a cat, together with another cat, can do, and the rock and the fire cannot do. Actually, this copying of information, many times, the copying of the genome, many times it's done with some errors, but actually those errors are very useful for Darwinian evolution, and we will touch on that soon. So if we continue this thinking, then maybe like the basic ingredients that we need for life, for minimal life at least, is that a system that can replicate, showing self-replication, so it has to self-sustain itself, but also being able to transmit information to the next generations. And then also it's something, if we want to then go from the minimal life to eventually life as we know it, to more complex life, it's also something that has to be able to undergo Darwinian evolution. And for that, the transmission of information actually has to be imperfect. So the idea of self-replication, which maybe exponential growth is like one hallmark example of it, but of course there are other ways to do it. Unfortunately, these days with the corona and infection, and infection number is just too real, right? Because we think that we have, a cell or one infected human, and then it can transmit this infection to others, and those others can transmit it to others, and so on and so forth. So this sort of replication. So a cell divides and gives rise to more cell, and those daughter cells divide again and again. Eventually, we have this population, but actually, because this division is many times imperfect in terms of the content of the transmission of information, then there is some variation in our population, and Darwinian evolution works on this variation in our population. So actually those variations are the base for Darwinian evolution. So let's think about the following very simple example. Let's think about a parent. In this case, it's a giraffe, right? And a giraffe is very tall, so it can eat leaves on the top of the trees, and therefore it doesn't have to compete with all the other animals that are eating leaves on the lower parts of the tree. So maybe it has less competition. But then this parent, because it is eating good enough, it is able to reproduce, and it is producing 3 children. So the first child, one of the children is actually of similar height to the parent, and so then it can compete. It competes over food with the other giraffes. But another child is actually a bit smaller, and this child is now competing with the other animals that the other giraffes are not competing with. And because of that, maybe this child is then not able to eat so much leaves, so much food, and then because of that, it is maybe not so well fit that it doesn't give so many children. And actually this child, the third child, which is a bit taller, now actually has basically no competition because it is not competing with all the giraffes that are tall but not as tall as this child, and there is not so many other creatures that eat leaves that high. So because of that, because of those small variation that gave this something that apparently gave it an edge, this child can then eat and consume much more food, consume better food, and therefore produce more children. And then eventually all the children will be taller and taller. This is, of course, a highly simplified example, but this is just to say, what do I mean when I talk about Darwinian evolution? So when we try to take this for the origin of life, and please take this picture as very crude, it's not very exact, we think of the origin of life as a continuous transition, not a jump, but a continuous transition from no life that starts with this prebiotic soup and ends with life as we know it, which is here symbolized by Luca, the last universal common ancestor. And like I said before, what we're looking for, or specifically what I'm looking for, is a chemical system that can give rise to replication and evolution. And I also tried to sort of try to connect it with some of the other talks that you had and mentioned protocell. So it's unclear exactly what is the stage of the protocell, but you can think that a protocell is something that is coming right after we have a system that is capable of replication and evolution. If a protocell is something that is very similar to present-day life, like it must have a membrane and it must have sort of like an RNA and maybe other stuff in there that resemble present-day life. So then the end of the origin of life is symbolized by Luca. Luca is the last universal common ancestor of life as we know it. This is this organism, this highly simple cell that sits at the base of the tree of life. Of course, there can be more ancestors and more stuff happening before Luca, and this is more stuff that's much more interesting. This is where it might be related for the origin of life. And one way that we can understand about Luca is by analyzing the genomes of the creatures around us and doing also sort of bioinformatics and gene comparison. And so we can come up with the minimal gene set that you need that is shared across all the life. And this might be the minimal gene set that Luca had. But actually, as simple as it is, and it is highly simple, and this minimal gene set is basically just encoding for just the replication, the transcription and the translation machinery and nothing more. But as simple as it is, and I think it's something in the order of only 100 genes, this is the result of this analysis. As simple as it is, it's still something that is highly complex just to form on its own. So we're looking for the series of steps that maybe eventually led to Luca. Now, the beginning of the origin of life, we begin with this early Earth, young Earth that had all the right ingredients for life, but it was not alive. And we like to call this sort of the primordial soup. So it had water and it had lots of simple molecules like sugars and lipids and all these sort of gases, but it was not alive. And we can know quite a lot about the state, the condition of early Earth by, for example, analyzing the content of old meteorites and seeing what is their chemical composition, or analyzing all the rocks that we find on Earth. Another thing that we can do, and I like to bring this example because it's a very famous example, we can sort of do experiments that try to mimic the conditions on Earth and try to see what sort of molecules can be formed in those conditions. So one of them is this Miller-Urey experiment, I think from the 50s or 60s. And what they did is actually they tried to mimic this condition when you have, like, on one hand, you have a volcano that is like generating a lot of heat, but then also occasionally you have this lightning, this electric discharge. And this is like how their experimental setup looked. And what they did is they mixed gases according to the composition of early Earth atmosphere. So they mixed ammonia that gives you a nitrogen atom, and methane that gives you carbon, and water gives you the oxygen, and hydrogen. So these are like the atoms that are highly abundant in life. And they mixed them all together, and then they subjected this to cycles of heat and also cycles of electric discharge. And then this was very nice because what they observed is that actually after those cycles, you see the emergence of simple chemical and biological molecules. And I'm talking, for example, molecules like lipids that are needed to make the membrane, or sugars, even simple amino acids, and even a precursor for nucleic acids. So it's very nice because what you saw is that in the conditions on early Earth, you can have the formation of molecules that are relevant for life. But I want to emphasize this still doesn't answer the question, because the question that we are interested in especially from the point of view of chemistry, is how life started, how all these molecules rearranged together. So we need to think about how all these molecules came together to give rise to self-replication and Darwinian evolution. So when we look about these two characteristics of life, then we can sort of think, oh, maybe we have a problem of the chicken or the egg, like which came first. And I'm deliberately not saying which is the chicken and which is the egg. But actually, my view is that it's not that one came before the other, but actually they are coupled. So we are looking for a system that gave rise to that is able to self-replicate and evolve in the same system, in the same unit. So we're sort of looking for something that I like to call the CHAG. Not a chicken, not an egg, something that combines them both. So what I'm going to do now is just to wrap up the sort of this part of the talk, I will actually discuss a candidate for a CHAG and another candidate for a CHAG, and then I will say a few more things about my own research. So we actually have an interesting candidate for the CHAG, and this is related for this concept that I think you heard in the first talk by Tony, the RNA world. And the idea of the RNA world is that it is drawing from the central dogma of biology. And the central dogma is that you have one molecule that encodes for the information, and then this information is, with the help of another molecule, is then eventually transmitted and converted into proteins. And proteins then fold, and they are the enzymes, they do all the work in the cell, or most of the work in the cell. So likewise, the idea of the RNA world is that you don't start with a DNA, but you actually start with an RNA. But then this RNA somehow has to both hold information and fold to do some activity like proteins. Actually, I can understand that there is a lot of reason in this RNA world hypothesis, because we know that RNA contains information because it copies parts, transcribed parts of the DNA. And then it was also found that some RNAs can actually fold and like a protein and maybe do very simple activities, like they can cleave themselves or something like this. So this is very interesting. So thinking about an RNA as an enzyme, the sort of a ribozyme, is very interesting because it has lots of relevance for the origin of life. But still the question is then, how do you form the first one? And I will not bore you with the details, but actually this problem of the RNA world assumes quite complicated initial conditions many times. people have been shown very nice work that sort of you can take RNA and then you break it apart, and then you can see that those parts can come together and replicate themselves and do very nice things. But I think that the challenge still remains that how you go from the very basic ingredients, how you can form this RNA that can then replicate itself and evolve. And this is still an open question. I want to bring it here that it's been several decades that this concept of the RNA world has been out there and people are studying it and people are making very nice progress. But also I wonder why after so many decades, so much time and effort of people, still we are nowhere close to solving the question of the origins of life. And so I would like to suggest an alternative. It's not me, this has been suggested long before me, and this is the idea that maybe the answer can come from chemistry. Now, we focus on chemistry not as the steps that we need to synthesize this or that molecule, but rather how can chemicals interact, simple molecules maybe, interact together to give rise to emergent properties. And we know that when you take simple ingredients and you let them interact together in sort of a network, so to speak, then you have emergent properties. And maybe the nice example is the brain. So if you take a neuron, or neurons, individual neurons, but then you let them interact in this neural network, then you have an emergent phenomena, which is the brain, which is conscience, which is why we are all here together. And likewise, the field of systems chemistry is going beyond looking at individual molecules, and we're looking at mixtures of molecules, of networks of interacting molecules, and we're looking trying to figure out, in some parts, what are the emergent properties of such systems, something that the individual molecules cannot do together, and we cannot have predicted it just by looking at the individual molecules. And maybe some of those emergent properties eventually can help or to lead to life as we know it. And within that, I'm more focused on just any chemical system that would give rise to replication and evolution. So now I want to transition, maybe still keep it more on the conceptual level, but transition more to my own work. But before that, I will just say that actually there are several candidates. They have been discovered in the last years, more than the last years, several candidates of chemical systems that can show very interesting things. Some of them even show replication. And I'm interested in basically any chemical system that can give rise to an emergent function. Maybe this emergent function is also Darwinian evolution. And I'm not so much focused on trying my own research. My own specific research is not so much focused about trying to figure out how we can have the specific structures that are needed for present-day life, like how can we have proteins and RNA and DNA, and more trying to see by borrowing from evolutionary biology, how can we understand evolution can happen in chemical systems, if it can happen. So one very nice example of a chemical system that shows self-replication actually comes from the University of Groningen, from the group of Sebring Otto, that I'm actually hosted in that group. And what you see here is that, I will show you a bit more about the specific molecules that we are using. What you see here is that when you begin to run the reactions, then you have a long period of this lag phase. And even though you see a little bit of the emergence of what would be the replicator, at first it doesn't replicate. And only once there is enough of it, I'm telling you now the secret, only once it is able to self-assemble, only then it can then begin to actually replicate and make more and more copies of itself at enhanced rates. And then we see this exponential growth. And then eventually all the mass in the system resides in this replicator. But this is not enough, because showing exponential growth is not enough to show that this is replication. You have to ask questions about templation. So what we can do to see if the replicators can indeed template is we can take a small sample of pre-existing replicators, and we can repeat this experiment from the beginning, and we see that once we seed the new system, the fresh system, in those pre-existing replicators, then first of all, we immediately get the replication. We immediately get the exponential growth. And also it is happening slightly faster in the non-templated case. So this really shows that what we have here is a replicator. Basically, the way the system works is that the self-replication is driven by self-assembly. So we have this building block, and in the head, we have two tiles. And eventually, what can happen is that with those two tiles, we can form disulfide bonds with other building blocks, and we can form this sort of macro cycles. Here, you see the hexamer. It turned out that actually hexamers like to stack on top of other hexamers. So eventually they form this sort of fibers. And it takes a while before the first hexamers form, and it takes a while before they can then stack on each other. And this is why at first you see the lag phase. But then once we have those fibers, they can get longer and longer, and more hexamers can stack on them. And if we now constantly steer the system, which is something regularly do in the lab, then some of those fibers can eventually break. And when they break, then they produce more fibers. And when they produce more fibers, then more hexamers can join. And this is actually the source of this exponential growth. I will give some more details, more reference about this later, that you can go and check more details. Actually, it's not just that. Because if we now go and we now have mixtures of building blocks in this system that I showed you, then what we have is something that might seem like evolution. So if you go then and follow the fate of this mixed system, then first of all, what you see is that they emerge in a very specific way. Because first of all, you have the emergence of the red-rich replicators, the rings that can replicate. And then this triggers the emergence of another subset of replicators, just a second, of another subset of replicators now, the blue-rich replicators. And actually, you can do further tests and confirm that what happens is that first the red emerges and then the blue emerges out of the red. And this is very nice, because what we see here is that maybe this is something that resembles the way that diversification has occurred in the extended history, evolutionary history of us, that led to the generation of the tree of life that we know. So this is a very nice system, and this is why I chose this system as an archetype to try to understand evolution in chemistry. I'm taking more of the sort of the networks approach. And when you think of a network, you can think that this is like the internet. So each of those circles is a web page, and the arrows between them are links between the web pages. So thicker arrows can mean more links, or links that are more central. But this can also be an example of a network of chemicals. So then now each of those circles is a chemical, and maybe the arrows represent how well those chemicals can self-assemble together, how well they can interact together. So if that's the case, then we can sort of look at two general examples. Like one is this more connected network, and one is this more self-centered network. And now this is probably going to be the last question that I will ask you, is which one do you think is better? I'm not telling you better for what, just which one of those networks is better. Is it better to have a more connected network or a more self-centered network? So here's the thing. If we are focusing only on a replication, the self-replication, then maybe the more self-centered system that is like this is better at this. Because, for example, as soon as we will start to have yellows, then because yellow tend to attract more yellows, then this attraction will be very, very efficient. And then eventually all our system will be yellow. But then if something happens, which this is the case in evolution, like the environment changed somehow, and then the yellow molecule overall is just not being formed well enough, or it's being washed away for some other place, then actually this system that only had the yellow then dies out. And this is not so evolvable. However, in the more connected system, maybe the replication process takes a bit longer. First, you have to recruit the gray, and then the pink, and then the blue, and then the yellow, and then the green before you can even recruit another gray. But this system is more evolvable, because at least it can respond better to changes in the environment. Because if the yellow then get depleted from the environment, then the system can still continue the cycle, but now it is doing a less efficient cycle, maybe with the red. Or we can cut this green to blue transition. Now maybe it's not so efficient, but at least it's happening. And this is basically the concept that I'm working on. So I'm trying to steer chemical replicators using insights from biological replicators. And I'm doing a lot of mathematical analysis and computer simulations. And in the example that I showed before of that chemical system, the question can be, for example, what do we need to do? What do we need to change in this dynamical system in order, for example, to get selection just for one type of replicator or for one group of replicators or one group of replicators. But you have to remember that actually these systems are, at the moment at least, highly unpredictable, and sometimes evolution sucks. Sometimes you can go from a dinosaur and you start to evolve, but then eventually you end up as a chicken, and sometimes it can be the other way around. So these are the messages that I hope that I was able to transmit to you. So how can we think about the origin of life, about prebiotic evolution, about chemical evolution, and that this is not just random concepts, but actually something that people are actually doing these days in the lab. I will maybe post on the chat or maybe on Twitter. I will post on Twitter these few references that you can go and access to get more details about what I was speaking. This, I think, A Simpler Origin for Life is a wonderful paper by the late Robert Shapiro. And if you want to know more, this helps really understand the idea of prebiotic evolution and chemical evolution. And if you want to know more about the details about the specific chemical system that I just shown you, the one that they formed those fibers, first of all, there is this nice YouTube animation that explains the process. But just if you want to get more, you can check out these references. And if you want to know more about my own research about chemical evolution and the chemical networks, then also you can check out this mini review. So with this, I want to thank all of you who took the time and come to listen. And also thank you for the organizers for making such a nice event happen. And I'd be happy to answer questions and discuss now.
Thank you so much for being with us today. So now we'll be moving on to the Q&A session, of course. So our first question is, maybe that's an absurd question, but if the protocells only have RNA, could viruses also be the origin of life on Earth?
Well, there is no absurd question. So if the protocells only have RNA, then I'm not sure if this is something that can be considered as a virus, because virus is something that is like falls a bit outside of our definitions of life, because virus cannot replicate themselves on their own. So if the first protocells only had the RNA, maybe they had ribozymes, special kinds of RNAs that can do some activities, but of course now you all know that this is not the answer. And then I'm not sure that they would be considered as a virus.
I understand. So our next question is, do you think ribosomes could be the first molecule that started life?
I think that ribosomes are extremely important, and look, there is no, you cannot argue with the fact that they are needed for life, because they are here now, luckily for us, because yes, of here. But the question is, what was the first molecule? I do not think that they were the first molecules. Unfortunately, I also don't have a good answer. I think that maybe some sort of a chemical system somehow was able to replicate and evolve, could maybe eventually give rise to something like a ribosome. But I'm also recognizing that on one hand, I'm saying that ribosome is not the answer, but I'm not giving you any good alternative. This is the case that is now in the research. You don't always have the right answers.
Okay, so maybe connected to this, if we can solve the mystery of the origin of life one day, do you think that we will be able to create a living organism using synthetic biology?
Yes. I mean, already now where people are doing amazing things with synthetic biology, you know, we can take cells, we can take E. coli and modify it to do quite a lot, sometimes even things it wasn't meant to do. Mm-hmm. So if we can understand how like to form life from scratch, then it is a possibility that using synthetic biology, we will actually be able to go and to mimic this process. We should just remember that even if we find how to create life out of nothing, because we don't have direct evidence for the origin of life, maybe there is still the possibility that even if we create life in the lab, this is not how it actually happened. Because until we have like a time machine and we go back, we cannot really know what happened.
Yes, definitely. I did not think about that before. Like, if you found how life happens in any way, we don't know the original way, and we need an evidence for that. Yes, yeah. Definitely. Eda Düzgün asked, and when we find the origin of life, in what ways will this benefit us? What do you think about this?
So so first of all, I think just by satisfying our curiosity. But I understand that often people want something more. So if we understand how to create life, maybe this can help us better understand life itself. So for example, here just the previous question was about synthetic biology. So maybe if we can understand how to create life from the very basic ingredients, from non-living ingredients, then maybe this will give rise to a new field of biotechnology that we're just not aware of now. But also, if we understand how life started, maybe exactly how simple life operates, this can help us guide our search for life elsewhere in the universe. Because now we're trying to search for life elsewhere in the universe by using signs of the life that we know, which is us. Maybe this can help us either specify or broaden the search.
Also, in the last couple of years, I've searched about this a lot, and people are trying to be integrated in technology and biology together with like biohacking and everything. And do you think we talked about evolution a lot? So I'm gonna ask about this now. So do you think people trying experiments out of official labs is affecting world's evolution? How can I say this? World's evolution process.
That's an interesting question. I think that this is beyond me, because evolution is such a complicated thing that to try to think about it in a simple system like a cell is already hard enough. So they try to see how it goes to combine it with the population and with humans, where it's always hard to talk about evolution in humans. This is just something that I don't think I can answer.
I understand. Thank you so much for coming and talking with us today. It was such an interesting topic for me, and I'm sure, as I see from the comments, everyone enjoyed so much. So now we'll be moving on with our break, but would you like to have some last words?
No, thank you. I enjoyed giving this talk.
Okay, thank you so much. Hope to see you again in the future. Thank you.
Bye-bye.
Bye.